Very welcome, everybody. A few people are still finding their seats. I'll just take the opportunity to introduce myself. My name is Magnus Lundgren. I'm head of the SciLifeLab Ancient DNA unit, one of the partners organizing this meeting together with the Center for Palaeogenetics and the National Genomics Infrastructure. For those of you who are online or would like to ask questions through our online interaction tool, you can use the software called Slido. There's a link distributed, or you can just use the QR code shown here. So with no further ado, I leave the floor to Anders Götherström to introduce the meeting and the field of ancient DNA research. Thank you.
Thank you, Magnus. Let's see if we get this one to work. Yeah, thanks. Thanks for letting me start off this meeting and say a few words about the history of ancient DNA. I am from the Center for Palaeogenetics. That's where I work. The Center for Palaeogenetics, it's a fairly new facility. We started operating in 2020. When we started, we were 16 people moving in there, and now we're soon going to be 45. It's a collaboration between the Natural History Museum and Stockholm University. A nice place to be, we think, who are working there. So, ancient DNA. I started in the field back in the 90s, and when I started, this was one of the first things I read. It was from the it was from the New York Post. Somebody worried about somebody else weaponizing dinosaurs. This is back in the 1990s, in the early 1990s. And if you go out and look at what we actually had back in these days, there were a couple of publications. Mark Thomas and Erika Hagelberg had 375 base pairs of cytochrome B from 2 mammoths, and there was some ground sloth work and some other stuff. A few steps away from weaponizing velociraptors and Tyrannosaurus rex, I think. But we have this relation with the media. We are working with materials that people think is interesting. We are working with events that people like to read about, and we are working with the molecules of life. So we do get a lot of space in media. And a couple of years ago, Elizabeth Jones wrote a thesis about that, and she turned it into a book. For us who's been in the field, it's kind of fun to read. It's basically the first 30 years of ancient DNA. If you're doing a brief history of the field, you can set it up over technical advances, and there are 3 that affected the field in one way or the other. The first one is the PCR, of course. I always used to cite a paper from 1986, but apparently it was invented 83 when I checked it out, and it got the Nobel Prize in 93. It was, most of you know what it is, and those who don't know will probably not be particularly interested. So just think about it as a way of mass copying DNA using the system that has evolved through life. And we could use that to do a few things. There was a couple of early papers based on PCR. There was the identification of some murder victims of Josef Mengele, and also Svante Pääbo, back in those days, he was exploring the molecule and trying to work out what ancient DNA really was. We were still just looking at it as DNA, as any kind of DNA. A real game changer for us was the silica purification. In molecular genetics, it just became convenient in general. For us, it made a big difference. Using the old techniques for purifying DNA, using phenol chloroform, we were co-purifying a bunch of humic acids and fulvic acids and stuff that more or less spoiled any possible practical analysis we could do downstream. So this, by binding the DNA to solid support and washing it, it turned into something workable. And for us, it made a big difference. It's after this we're starting to get things like this. The identification of the royal Russian family, for example, and the first Neanderthal papers were coming out in this era. In Stockholm, also all the domestication papers. This is one by Gregor on pigs. A lot of these studies back in these days, how, when, and where were various domesticated. In Stockholm, we were doing Neanderthals as well, the second Neanderthal sequence, and Love was starting to explore the circumpolar fauna. All this came with the silica purification. The next advancement, and the last one so far, is of course the sequencing strategies, the sequencing chemistries. We had been taught how to collect DNA information using Sanger sequencing, and then this stuff came along that sequenced DNA 1,000 times, 10,000 times faster, and we had this massive amount of data. And this is when we started to get genomes. The first ancient human genome, the first decent ancient human genome from Copenhagen, and just a few months later, the first Neanderthal genome, the first decently sequenced Neanderthal genome. Yeah, this is when we're moving from mitochondrial DNA to genomic data, in Stockholm we're doing it as well, and together with Uppsala, working on migrations here. I'm an archaeogeneticist. Back in these days, the paradigm was that people did not move; they lived in the same place they always lived. And over the last 10 years, in a set of studies, we and others have been exploring mobility. And in Stockholm also, the group working with the circumpolar fauna was exploring that part further. Back in these days, there were like 10 genomes, now there are 10,000. It's moving fast. So if that was the past, where is the future? I personally think that this is the future of ancient DNA. Very old DNA. We have done a lot over the last 10,000 years. But what happened 100,000 years, a million years ago? Microbial DNA, it's not only humans or mammals who has their DNA in the skeleton, it's also the microbes, and we can work with those. And also the stuff that we find in the sediments. We will hear about this over the day. We will hear Camilo talk about very ancient DNA. We will hear Zoe talk about microbes. We will hear Pete talk about sediments. And that is the field of ancient DNA to me. Thank you.
Thank you very much, Anders, for that nice introduction. And then we continue with the first keynote of today. So we are very happy to have Mehmet Somel here from the Middle East Technical University, who will give an overview of his research and the contribution of ancient DNA to that.
And I'm really honored to be here and give a talk on present our work. We've been doing in Ankara. Let me see. Yes, I think I can. Let me see if we can. Yes, perfect. Hello again, and thanks again. So we are a team in Ankara with focus in the history of our region, Southeast Asia, Anatolia in particular, and trying to use DNA to study human history. We're also interested in other topics like microbes, domestic animals as well, but most of the focus has been in humans in recent human history. And it's a collective of researchers working at two universities in Ankara, also including archaeologists and anthropologists in the team. And the bulk of our work in recent years has gone into understanding the Neolithic transition. That is the transition from the ancestral lifestyle of mobile hunter-gathering to sedentary farming, which eventually, as we all know, gave way to complex societies that we know of, that we're familiar with today, agricultural societies with states and class structures, so hierarchy and a lot of inequalities. So that was all possible through the Neolithic transition. And this switch from mobile hunter-gathering to sedentary food-producing societies that happened in many different parts of the world in different times over the last 10,000 years. But the earliest case, as far as we know, happened in Southeast Asia. And in this region, also called the Fertile Crescent because of its shape. And it was it was a long process. It took thousands of years of communities here changing their lifestyle. And in particular, the sequence in Southeast Asia follows this particular pattern of groups first becoming sedentary, building houses, permanent residences, also monumental structures like in Göbekli Tepe, and experimenting with plant cultivation, animal management, which eventually become domesticated. And then in the latter half of this period, and actually after 7000 BC, do we see full agricultural villages appearing, which then over a couple of thousand years into the Bronze Age become hierarchical in class-based societies. So the questions I'll be telling you about, we'll be talking about today are what kind of human mobility patterns accompanied this change, this switch, both during the Neolithic and afterwards, how human movement patterns changed over time. And also, briefly, we'll be talking about how human social structures, social traditions changed. So what ancient DNA might be telling us beyond what we already know from ethnographic and material culture studies. So with the question of regional human mobility, it's interesting because this, I told you this was a broad region where this Neolithic lifestyles emerged. And in different parts of the region, we find new rituals emerging, new technologies. As I mentioned, there are different ways of cultivating plants, managing animals and stuff, different types of architecture. Some of them remain local, but many of them spread over time. They're shared across different regions. And a question that has been obsessing archaeologists, of course, is how much this spread, these cultural sharing diffusion is happening through human movement, or perhaps even replacement of populations, like what we've known in the case of European Neolithicism, where farmers from the Middle East were moving into Europe and setting up farmer colonies there, basically. Whether the same type of dynamics were happening also in Southeast Asia, or was it basically groups sharing their knowledge, their new cultural traits with their neighbors? And both other groups and ours have been tackling this question for some time, also through a lot of collaboration with Anders and others in Sweden, the researchers in Stockholm and in Uppsala. So Reyhan, who did a PhD with us, and now she's working with Anders, she produced a couple of years ago new genomes from two sites from central Anatolia from the Neolithic period, early Neolithic Ashikle, pre-pottery Neolithic, so-called before full-scale agriculture, and Çatalhöyük, a full agricultural village from the later pottery or ceramic Neolithic. I might just point, not this one. Ashikle and Çatalhöyük, and she compiled these genomes She joined them with published genomes also from the region, including genomes from Paleolithic, so pre-Neolithic groups published before from Pınarbaşı or from the Natufian site here. And one question was, how much population structure do we see in this region? Are they all homogeneous or are they very differentiated? And indeed, as others had also been observing with more limited data, what we saw was that there is quite strong population structure in that Levantine groups, they tend to cluster together except for this single outlier from Kvaroj. Anatolian groups also cluster, and this includes not just Neolithic Anatolians, but also the Epipaleolithic Anatolian a couple of thousand years ago, which suggests that these populations were mostly before the Neolithic and after the Neolithic. There was a strong continuum, population continuation in Anatolia as well as in the Levant. And Zagros populations, also Neolithic groups here appearing distinct. Despite this overall pattern of population continuity within the Neolithic in Southwest Asia, so regional populations appearing to have mostly remained local, there was also some signal of gene flow. Again, what Rehan was finding, and as others had also published earlier. So, for instance, when Rehan tested if early Neolithic Anatolians and late Neolithic Anatolians, if they're equally distant to other populations of the region, let's say Caucasus or Zagros, so eastern sources, she found that late Neolithic Anatolians actually are sharing more alleles with eastern sources than early Neolithic Anatolians were. So this suggests there was some amount of admixture, human movement from the east into Anatolia over time. But a key region in this in the Fertile Crescent that had a major role in development of new technologies and also domesticates has been North Mesopotamia, this region around the Euphrates and Tigris rivers. And this had remained unsampled until recently, genomically unsampled until recently. And that was partly because the climate here is much warmer than in central Anatolia, so although we had access to material, it was not as successful. Our attempts weren't as successful initially, but eventually we did start getting positive results. So one study site here is Çayönü. It's pronounced Çayönü. It's from it starts from the pre-Pottery Neolithic age, pretty early in the Neolithic, and is continuously inhabited, uninterrupted apparently, for about 3,000 years. Quite amazing continuity there. And it's also famous for the community changing its agricultural styles over time, quite dynamically experimenting with pyrotechnology, with metallurgy, trying, there's also evidence of domestication, attempts to domesticate sheep, pigs, and also bred domestic wheat. Wheat strains are found to be, are thought to have come from that region, so there was also plants domestication happening. A lot of also experimentation with different types of rituals. So they were burying their dead for some period within their houses. Then they also had the collective, some kind of collective burial building. So it was something called a skull building. So a lot of cultural experimentation there. And we a couple of years ago, we screened 30 plus skeletal material from this region. Many of them contained no DNA, and about 13 contained about 1% or even less, but with a lot of sequencing, we could get partial genomes that allowed us to do some amount of demographic analyses. So Deniz, a postdoc in the lab, she produced the genomes in also collaboration with Anders, and Ezgi, another young PI, she analyzed the data, and she joined these 13 Çayönü genomes with published genomes again from the region, chain is located here, the red star, and our samples were distributed around 1,000 years, around 8,500 to 7,500 BC. And so this graph is again showing it's a MDS plot, a summary of genetic distances, now including individuals. And we again see the same structure, it's just flipped. Here we have Anatolia, Levant, and Caucasus and Zagros genomes. And again, the Anatolians we see quite clustered, Levantines as well. And where Chayunu stands in this picture, when we added that data, we saw that it's somewhere in the middle, tending closer to Anatolia and Levant, so potentially sharing more ancestry with them. But also, its location in the MDS suggested it might also carry some Zagros-related ancestry. And when Izgi ran QPDM analysis to try to explain the Chayunu genomes as a combination of ancestry from different regions. She indeed found that about 50% of the ancestry could be coming from a more Anatolian-related source, and 30 and 20% of ancestry from Zagros and Levant-related sources, respectively. Of course, it's a very rough analysis, but it suggests that the population here was sharing drift with regional populations from across the Fertile Crescent. When this admixture, these potential admixture events actually happened, when this mixture, gene pool mixture arose, we don't unfortunately know yet. With more genomes, especially from earlier periods, we would potentially be able to know. With higher quality genomes, we would also have a better chance of identifying potential admixture dates. But at the moment, what we can say is that this was a mixed group. We could also use this data to answer other questions about to better understand the history of the region. So I told you earlier that we were seeing in central Anatolia eastern admixture. So from the early Neolithic to the late Neolithic, there was eastern related ancestry sources arising in central Anatolia. But what source was actually involved, we didn't know. And when we tested Chayunu and the Caucasus and Zagros as potential sources, we saw that the Chayunu genomes were a much better representative of a potential contributor than Caucasus and Zagros to late Neolithic Anatolians like Çatalhöyük and Barcın and Tebek Çiftlik. So most likely the eastern movement was actually originating from Mesopotamia rather than somewhere else in the Fertile Crescent or the Caucasus, which would geographically also make sense and is also consistent with some cultural affinities people have recognized between Upper Mesopotamia and Central Anatolia. But in later coming periods, in other Anatolian sites, we actually see higher affinity to Caucasus, so it suggests it could be some other eastern source continuing gene flow into Anatolia after the Neolithic. But the Chayunu, there was something else interesting in the data. The Chayunu sample, these 13 genomes, were not super homogeneous, and there was particularly one individual which looked like an outlier. It was in the MDS plot, it was shifted towards the Caucasus-Zagros group, as you can see. The lab ID was Chay8. And Izgi could model her genome as a mixture of the rest of the Chayunu genomes and about 20% additional Zagros ancestry. So it looked like someone with somewhat different ancestry from the rest. So was it just a passerby, a migrant who ended up dying there and being buried in the settlement? Well, it turned out she wasn't really an outsider. She turned out to be a 2-year-old infant, a female infant, and she was buried inside one of the buildings, underneath one of the buildings, which was a tradition of the time in many settlements, burying the dead under buildings. And she was buried with some other individuals, one of whom turned out to be a relative of the this Chayet girl. And by studying the kinship, by studying genetic affinities between these two, between pairs of individuals in our sample, estimating kinship coefficients using different methods, one developed by Thorsten's group in Uppsala and one developed by using some other approach developed in Copenhagen. We consistently found that these two individuals were sharing were genetically as similar as one would expect from approximately third-degree relatives. Could be great aunts, could be first cousins. One other interesting thing was that so we were finding this third-degree relationship on the autosomes, but when we checked the X chromosome, we were actually finding stronger similarity between the two individuals. And one explanation for that is that these two could be related through the paternal line. And Ezgi constructed a couple of alternative pedigrees and simulated data, and she suggested most likely this other related individual could have been a paternal great aunt of the infant, whereas the Zagros ancestry seems to be coming from the maternal side. That seems to be more likely, also because the infant was carrying a mitochondrial DNA haplotype, which was also an outlier in the Chernio sample, but was found in Zagros samples. So, but that's not the end of the story. There was something further interesting about this girl, which was the skull. So our anthropologist colleague Yilmaz, who is also working in Ankara, he identified a scar on the small girl's skull that he interpreted as a cauterization skull, so some kind of heat scar that had eventually healed. So she had survived this so-called operation, or this event, and moreover, he also identified patterns that were consistent with some kind of inflammation inside the skull. So his hypothesis that somehow this girl had some severe illness, and the community, they tried to heal it through cauterization, and she did survive the operation, but eventually she did succumb to, she did end up dying quite early. This is also perhaps not super surprising, this kind of intervention, because we also find multiple skulls in Cheyenne and in the region of trepanation, so hole drilling inside skulls that where people have survived after the operation. So there was a lot of experimentation with different medical procedures. Well, medical. So to sum up, we see large-scale population continuity through the Neolithic transition across Southwest Asia. So all this cultural change, it seems not to have come with large-scale population replacement, but more sharing of cultural items and also domestic species, accompanied by some amount of human movement, as also evidenced by this small girl, perhaps. I don't have time to go into that, but the intensity of human mobility also seems to be varying in time. So in some periods it's more intense, in some periods less, but it seems to be more or less continuous. And we also are finding evidence that North Mesopotamia was a key region here, which was both potentially demographically connected to other regions in the Fertile Crescent, the Zagros, Central Anatolia, and potentially also Levant, and it was also culturally connected. So that's fine, but what happened afterwards is also an interesting question. So how human mobility patterns continued to change after the Neolithic, when full-scale food-producing societies were established and they grew, and you got more complex, larger-scale organization, also novel technologies of transport, and so on. Trade routes being established, armies, invasions being organized. So this we know a lot, quite well from the archaeological record, that human groups were moving or being moved around, that there was long distance trade. But all these evidence, how it might have impacted all these processes, how they might have impacted the gene pools of the region, how they might have impacted diversity, that has been only only recently being tackled. And recently, a PhD student, she just finished, Dilek from our group, she combined about 400 ancient genomes from the region, spanning the whole Holocene, including new genomes we published from Anatolia, and that Anders and Ricardo also contributed from Stockholm, and also Heraklion, our collaborators from Greece, Heraklion also contributed. So it was a wide-scale effort to try to understand the history of this region through the Holocene by studying genetic diversity and how it changes over time. And one pattern that Dilek could recognize, directly looking at the data, within each region here, Anatolia, the Aegean, referring to Greece here, Iran, Zagros region, and also eastern Iran, the Levant, and the South Caucasus. Across these different regions of Southwest Asia and East Mediterranean, we see a relatively monotonous trend of increasing diversity within gene pools. So genetic diversity, as we measure by how much differences, genetic differences, two pairs of individuals are present in the sample over time. So time here shown on the x-axis. Over time, the genetic diversity patterns tend to increase in all regions. And one explanation for that is admixture. So alleles coming from elsewhere, from the region or outside. We'll talk about that in a second. Alleles coming from outside, making local gene pools more diverse over time. And Dilek also noticed this pattern in PCA plots, now separated in 6 different time periods. So initially, the different colors represent genomes from different regions of East Mediterranean and Southwest Asia. And around 10,000 years ago, the genetic differentiation appears quite large, but then over time, the gene pools become more similar to each other. They kind of converge in PC space. So there seems to be a tendency towards homogenization of these gene pools, at least initially, in the early millennia of the Holocene. And that had been, in fact, also observed by others as well, especially measured by this statistic called FST. So it's a measure of genetic distance between two groups. And FST, as you calculated early on with the genomes around more than 10,000 years old, is quite high, FST between regions. So each point here representing a pair of regions in our broader region of interest in Southeast Asia. So each point is a pair of regions, and FST over time it drops quite dramatically, suggesting that these groups are just mixing together. But there's one issue with this FST measure, which is that it calculates distance as a ratio between divergence, so genetic absolute genetic distances, let's put it that way, and but divided by diversity. So it's also impacted by diversity, which I just show you also changes over time. So an alternative, we thought that an alternative way to measure genetic distances could be 1 minus F3. So F3 is a statistic that you could use in a way that is also robust to genetic drift, so it would not be affected by population size changes, and just reflecting how much genetic distance you have between two genomes or genome samples in two regions. And if we calculate the same, if we calculate genetic distance now using 1 minus F3 instead of FST, we initially find the same pattern of decrease in genetic distance between regions from about 10,000 years ago to about 6,000 years ago, around the early Bronze Age. So this suggests that indeed the gene pools, as we also saw on the PCA, gene pools seem to be mixing together, so that would decrease their distances. But then in the latter half of the Holocene, after the Bronze Age, there is a tendency of increasing distances, now as measured by 1 minus F3, something that was opposite to the trend we saw with the FST. And this could be explained by gene pools, the regional gene pools receiving gene flow alleles from from other external sources, more distant sources, that would make them more divergent from each other. And this kind of increasing divergence over time, first decreasing divergence, first gene pools becoming homogenizing within the region, and then becoming more divergent, it would be compatible with also what we were seeing, we and others as well have been seeing at the level of ancestry components, as measured, for instance, estimated by QPADM methods. So in the first half of the Holocene, we see ancestry components mixing within the region, just like what I was showing you was happening in central Anatolia. So Zagros or Mesopotamia-related ancestry arriving in central Anatolia, so regional populations mixing with each other. But in the second half of the Holocene, after the Bronze Age, we see evidence of longer distance migration, gene pools in these regions being affected by different sources individually, distinct sources individually, which include, for instance, the Yamnaya affecting the Yamnaya migration. Oh, sorry, this is Turkish, but never mind. So affecting the Aegean or the Caucasus, but not Anatolia, whereas Anatolia being influenced by Central Asian gene flow and so on. So what we suggest was that there was a change in the dynamics of human mobility from early Holocene to late Holocene, especially with the Bronze Age period, that the scale of mobility, human mobility, although mobility was continuous, its scale increased dramatically after the Bronze Ages, which would make sense because the scale of human organization and also transport means, of course, changed quite dramatically during this period. One other interesting observation from this from the study was about sex-biased mobility. So this is not something super easy to study, but we thought we could tackle it by using information from the autosomes and comparing it with information from the X chromosome. Because females carry 2 copies, females and males both carry 2 copies of autosomes, and females carry 2 copies of the X and males only 1 copy of the X. If migration between from a region to another, if it only involves females, then you would have equal genetic change on the autosomes and the X chromosome. Whereas if it's male-only admixture, you would expect only half of the change on the X chromosome compared to the autosomes. And what Dept did was she measured genetic distances over time, so how much genetic change is happening in each region from one time period to the next on the autosomes and on the X chromosome. Comparing gene pools from different consecutive time periods. And so overall, there is a correlation. So the more change in the autosomes, the more change on the X. But there was this pattern that points that were from the earlier periods of the Holocene, so each point here representing a regional group, and different colors representing, different regions, and different shapes representing different time periods. And you might already have noticed that the early time periods, they tend to be below the regression line, whereas the later time periods, they tend to be above the regression line. And if we visualize the same data, just calculating residuals from the regression line, we find that we tend to find negative residuals early on in early time periods, so between 10,000 and 8,000 BC before present. And we tend to find higher residuals in later time periods. And one explanation for that is that there was increasing male bias in human mobility patterns. So either initially mobility was more female biased, and then this decreased over time, or initially it was more equal and the male bias increased, or both processes happening at the same time. So we cannot exactly distinguish between these two scenarios, but what we can tell is that female to male bias, it shifted over time towards more male bias. And yeah, that was also, of course, accompanying this pattern of more expanding range of human mobility. How much time? I have 8 minutes. Okay. Okay. And the last part of the talk is now going beyond large-scale human mobility. I'll be telling you about human social traditions and what ancient DNA is telling us about how these might have changed over time. So this is also an interesting question, because this dramatic change in lifestyle, it seems to have been accompanied with changing social traditions. Now, many modern-day agricultural societies, as we know them, they tend to be quite strongly organized around family lines, nuclear families, extended families. Also, many tend to be, not all, but many tend to be patrilineal and patrilocal. But both archaeological and ethnographic evidence is actually suggesting that the ancestral hunter-gatherer groups were actually not so much organized around family lines. So many groups with 20, 30, 40 members were not closely related. So hunter-gatherer bands were not composed of pretty closely related individuals. The postmarital residence patterns, so who once you have a marriage, in which group the pair stays with, was more bilateral. So the pair could choose the woman's family or the men's family to continue their activities. So there is this. There seems to have been a significant shift in how societies were organized through or after the Neolithic, and yeah, how this happened. So how Neolithic societies themselves were organized has been a major question. There's also the point that some archaeologists make that perhaps more family-based, more potentially nuclear or extended family-based organization might also have promoted the investment in buildings, in agriculture. So it might have a more family-based organization might actually itself have promoted Neolithic groups, Neolithic lifestyles being established. But of course, this is not possible to directly test using archaeological material culture data. What we just know is, as I was just mentioning you, from the ethnographic evidence that hunter-gatherer groups from the present day, they tend to not be so much organized along family lines. And for instance, horticulturalist groups, they compared to hunter-gatherers living in the same area, they tend to be more family-based and also potentially more patrilocal. And also, archaeogenetic evidence now is coming in, especially from Europe, where we see already by the late Neolithic and early Bronze Age, based on cemeteries and kinship analyses run on DNA data from cemeteries, that these groups tended to be quite patrilineal and patrilocal. So we tend to find adult males being frequently related, but adult females being seeming to have come from other regions, other communities. So this is, of course, two ends of the distribution, and what we want to understand is what was happening in between. And potentially Neolithic Anatolia and Southwest Asia could help. And we're extremely lucky there because there was this tradition I was telling you about of people burying their dead within buildings while the buildings were in use. So in Çatalhöyük, for instance, this is the reconstruction of Çatalhöyük house. We had people, both adults and sub-adults, burying under the floors. So what we did was collect data where we had multiple individuals buried in the same buildings or in the same spaces from different Neolithic Anatolian groups. And I'll just show you briefly the results here. In Çayönü, this is early Neolithic from North Mesopotamia, as you remember. We had multiple individuals buried in 3 buildings, and in all 3 we found relatives. So they were, not everybody had relatives, but quite a few had relatives in the same building. In Boncukluyuk from Central Anatolia, again early Neolithic, so pre-Pottery Neolithic for us, we had a group of individuals buried in 2 neighboring buildings. Again, we found relatives. Not everybody, again, was related. We had this interesting case of a perinatal baby buried with a young woman, but they turned out to be unrelated. But overall, in the sample, quite a few had relatives. Aşıklıhöyük, again, Central Anatolia, early Neolithic, quite a few close relatives, first, second degree relatives. But when we go forward in time, and we had two settlements where we had data from the later Neolithic. From Barcınhöyük, we had a couple of, sorry, this is now Çatal. From Barcınhöyük, we had these infants buried inside houses, sub-adults buried inside houses, and although we did find a couple of related pairs, now the relatedness frequency seemed to have decreased. They were, again, buried in the same building, but many did not have relatives that we could identify. And the same pattern in Çatalhöyük, again, later Neolithic, Central Anatolia. For instance, these 2 were not related. These 4 individuals were not related. There was 1 pair of relatives. So overall, the data is still, the data that we published yet is still relatively sparse, but there seems to be a shift in related patterns among co-buried individuals within buildings. In the early Neolithic, in the pre-Pottery Neolithic, it seems to be more frequent that people are buried with their close relatives, whereas in the late Neolithic, in Barcan and Çatalhöyük, this seems to have decreased in time. And with now a larger sample that is not yet published, about 100 genomes from Çatalhöyük, across 33 buildings, we find the same pattern. In fact, so this is a sample that spans around 1,000 years. And over time, just within Çatalhöyük, we see a tendency of decreasing relatedness among individuals buried in the same building. What could be the reason? We don't actually know, but one possibility is that this co-burial of unrelated individuals and potentially biologically unrelated individuals living together, or children being somehow brought together in houses. It could, the archaeologists are interpreting this as a potential way of consolidating community ties in a period when society was, there was actually a tendency for disintegration, more private storage, not still egalitarian traditions continuing, but at the same time, society potentially being more atomized, and the idea is that this kind of new tradition of mixing up families could be a response to that, to those stresses. Of course, this is yet speculation, but it is. We do seem to be finding a new tradition arising in this period, in the later Neolithic. I didn't have time to tell you, but it also seems as if patrilineal burial traditions, they appear to have also arisen later than the Neolithic. So we don't see very strong patrilocal, patrilineal patterns in our sample yet. But this is still data we're working on. So I need to thank many people, both having shared their material with us and having discussed and interpreted our data, our archaeologists and anthropologists colleagues, people who, of course, worked in the wet lab, and also people who analyzed the data. Also, Anders, for our collaboration has been very central to our work for last 7-8 years. And thanks a lot for having me.
Thank you very much, Mehmet. That's a fantastic presentation on a key time period in human history. We can take one question from the audience. Is there anyone who has a question at this point? While you're thinking, we have a very specific questions on the web, and that's on your FST analysis. So they was wondering if you used WC or Hudson for your FST analysis.
No, I think it's the WC. So this was the WC. Yeah.
Okay.
We're cooking.
Is there any question in the audience at this point? All right. Thank you very much, Mehmet, and make sure to grab him in the break and ask more questions about his fantastic work. Thank you.
Excellent.
Now, the last presentation before the coffee break, we have Carina Schlebusch from Uppsala University, who are going to talk about African history, inferences from modern and ancient DNA. So please, Carina.
Thank you very much, Magnus, and the organizers for organizing this nice symposium where we're all going to talk about ancient DNA, and we use ancient DNA for various things, for inference of human history, looking at viruses, looking at plants. And ancient DNA is a tool that we use more and more, and together with some of the other disciplines that investigate human history, like linguistics, for instance, and archaeology, we are starting to find out quite a lot about how humans populated the planet and find details about human history and in migrations in the past. So, for instance, I don't think, oops, looking for the pointer. Let's see. Oh, yes. For instance, here you can see quite a lot of detail here in the European and West Asian region when you look at those arrows and so on. And many of these fine-scale inferences we have because of ancient DNA, because of the ability to sequence humans in the past and to infer all of these overlaying migrations in this region of the world. And when you just look at this picture, for instance, for Africa, it doesn't look so clear. It looked quite undefined, actually. But we're also learning more and more for African history. For instance, what we find when we look at African modern and ancient genomes is that we have two stages in African history. The deeper phase that is defined by when looking at hunter-gatherer genomes and also the genomes of ancient DNA individuals. And then we have a later phase defined by large migrations of farmers across the continent. And then we have the second phase in the Holocene where we have this larger scale migrations that we actually clearly see when we're looking at both modern day and ancient DNA groups. And as I said, the ancient migrations is then defined by looking at hunter-gatherer populations together with ancient DNA. And then even though we clearly see signals of these later migrations in modern day genomes, ancient DNA can be used to give us finer scale details. And this was a review that we did in 2020, and Mario was the first author. Mario's here today. So we took at that stage, up until that stage, all of the ancient DNA studies that have been published for Africa, and we analyzed it together with modern day genomes. And so far, not so many ancient DNA studies have been published for Africa. It's still Africa ancient DNA in Africa is still in its baby shoes. The first genome only been published around 2015, so 7 years of ancient DNA history, if you can call it like that, for Africa. But we have these 11 studies already, and they are starting to learn us a lot about African history. So, for instance, when we did this analysis, when Mario did this analysis, and he analyzed the modern day and the ancient DNA genomes together, so the ancient DNA genomes are these diamonds, and then the modern day are these dots. When we analyze it together, this is the two-dimensional genetic structure that we get for Africa, more or less. And you can see there's some correlation to geography. About 30% of the genetic variation is explained by geography. And these are North African genomes, these are West African, these are the East Africans, and these are the Southern Africans. So at least some correlation to geography. However, if you limit the data set to only hunter-gatherers, so ancient DNA from hunter-gatherer contexts, and also modern-day hunter-gatherer groups, you see quite a lot of variation being explained by geography, around 77%. And then linking back to this inference that we see in the past, we see how hunter-gatherers and how the populations living across the continent is defined by geography with these gradients of relatedness between different groups. And you can see the colors here overlapping the rainforest hunter-gatherers in Central West Africa, the East African hunter-gatherers, the South African hunter-gatherers, and there's quite a large correlation to geography. And this is in contrast with these large-scale migrations that we see later throughout the continent. We then also represented in an ancient DNA study that we did in 2017, we represented the relatedness between the different African populations in the form of this tree. So this is a population tree, and it shows the general average relatedness between different African populations. And here we see the first split in the tree is between Khoisan hunter-gatherers from Southern Africa and the rest of humanity. Then we have a split between the rainforest hunter-gatherers and East and West Africans. Then East and West Africans split from each other. And here we have the out-of-Africa migration as a subset of genetic variation in Africa, together with a big bottleneck where a lot of genetic variation was lost. So when you look at genetic variation in African populations, just looking at this deep structure, it is quite expected that you see quite a lot more diversity in African populations compared to non-African populations. And that is what we also see in African populations, this large diversity compared to non-African populations. But what does this tree mean? Actually, when we look at this tree, should we interpret that around 300,000 years ago, we were all one population in Africa, and then we split into these different population groups? And actually, no, this is just a tree representing the average relatedness of the different groups across the continent to each other. There are many different models that can underlie a tree like this. For instance, this model is called the isolation by distance model, and this is also a model that can underlie a tree like this. And in this model, there's many different populations across the continent existing in parallel deep into the past, but they are exchanging gene flow with each other in a stepwise manner. This could be continuous gene flow, or it can be like pulses over certain time periods, maybe when the climate permits, or whether groups become in contact when there is instances of gene flow. What do we see when we look at the paleoanthropology record? For the paleoanthropology record, we see from a period from 700,000 years ago to present that in different parts of Africa, in North, East, and Southern Africa, there is people occupying these regions. And it seems that there's a gradient of development where humans develop from archaic forms into more modern-looking forms. And this is anatomically forms. So when you look at the anatomy of these humans, these ancestors, we see gradients in North, East, and Southern Africa of development into more model-looking forms. And in this time period, when the populations start to diverge from each other, around 200 to 300,000 years ago, we see in the different parts of Africa, we see transitional humans. So humans that have some archaic features and some modern features, seeming that these humans were switching to modern anatomically modern humans in different parts of the continent. And this is also then what the paleoanthropology community are thinking, that there is maybe a pan-African origin of modern humans and not just a single origin. This is a very nice review article that summarizes the different possible models of human origin. Here is the single origin model, where one group expands and replaces all of the other groups in Africa. This is this long-time connectivity model that I just talked about, isolation by distance model, where there's stepwise gene flow between different populations. And this is a kind of intermediate model where one group expanded and mixed with the other groups in the continent. So these are possible models that we need to test to see which one is more likely for African data. Some interesting things that the article note is also that there could be archaic admixture. And this is very interesting because we have seen some evidence of archaic admixture already in some genetic studies. So it looks if some archaic groups might have mixed into modern day groups in Africa. We're not sure because we don't have African archaic genomes. So these populations are called ghost populations. And I see that Tom has a talk on ghost archaic admixture a bit later today. So I'm looking forward to that. But all of these studies infer that there might be evidence of deep coalescences in the genomes of Africans, indicating that there might have been archaic admixture in some African groups. So what that would mean is, so here we have a split between the Southern African hunter-gatherers and East and West Africans. So this is all modern day populations. But then this is an archaic genome that might have sort of have a divergence in between Neanderthal genomes and modern day groups, so a deep divergence, and this genome is then mixing back into some of the African groups. So we've seen indirect evidence of this in the genetics, nothing very certain yet. What we see in the paleoanthropology record, there was definitely opportunity for archaic humans in Africa to mix into our lineage. For instance, here in Southern Africa, we have this very archaic looking Homo naledi that was living alongside this transitional Homo sapiens. So this might have been among our ancestors. It was already transitioning into a modern looking form, while this Homo naledi was a very archaic looking form, but they were living at the same time on the same landscape. Maybe this was too archaic, maybe it's too archaic. They were too different to mix. Was there something that were more likely that have mixed with us? Yes, there was also this broken heel or Kapwe population or individual that looked a bit more modern. It's classified tentatively as Homo heidelbergensis. And this Homo heidelbergensis is the common ancestor, or many people think it's the common ancestor of our lineage and Neanderthals. So we've mixed with Neanderthals. This individual lived more or less in the same time period as these two individuals from Southern and East Africa. Definitely they would have met each other. Would they have admixed? We don't know. We see some lineages that have deep coalescences in African sequences. This might be one of the populations that mixed with our ancestors. But nothing is sure yet, and it is going to be very difficult to really show that archaic admixture happened in Africa without getting an archaic genome. Because with Neanderthals and Denisovans, it was a to and fro whether we did admix with them or not, or at least with Neanderthals. And until we got a Neanderthal genome, we couldn't really say whether we admixed with Neanderthals or not. So it will be interesting to see what is going to happen for Africa. Now I want to highlight a few of ancient DNA studies that contribute to these Holocene migrations, so these large migrations that you see on the map here. First, I want to start with the North African. So today, people living in North Africa, we know that when we look at them genetically, they look more like non-Africans and not like sub-Saharan Africans genetically. So then the question was, when did this back migration into North Africa happen? And was it actually a back migration, or was it out of a sub-Saharan Africa and North Africans always look like non-Africans? So this is a question that is still pending, but ancient DNA study from the Max Planck Institute actually contributed at least some information to us by sequencing the genome of Tafferald, a 15,000-year-old individual. And when looking at Tafferald, we saw that Tafferald already had around 50% of non-African DNA in its genome, and then 50% was African. And this African part was more or less half East African and half West African. So we know at least 15,000 years ago, half of the genomes of North African people came from non-African sources. And this we know because of ancient DNA. There are more studies, also a study that we have out on bioarchive that showed there were continuous admixture actually from the Arabian Peninsula and also from the Levant into Northeast Africa. So more instances of back migration into North Africa. But the one I want to talk about a bit further now is this migration of East Africans into Southern Africa, also introducing herding practices into Southern Africa. And what we see and what we saw, we and others saw when looking at ancient DNA, is that we actually see evidence of this migration of Northeast African groups into Southern Africa. When we compare ancient DNA individuals from pastoralist context, this 1,200-year individual with individuals from hunter-gatherer context, and here you can see, 5 minutes, good. Here you can see these 2,000-year-old hunter-gatherer genomes are this pink color here, but then this pastoralist individual have a genetic component that is similar to the pastoralist living in Tanzania and Kenya, indicating this migration of pastoralists into Southern Africa, introducing pastoralist practices. Then the last example I want to discuss is maybe one of the biggest migrations in the Holocene in the world, is the migration of Bantu-speaking people from West Africa to the rest of sub-equatorial Africa. For a long time now, we know that this was endemic diffusion, so people moved together with their language and their culture across sub-equatorial Africa, and they replaced all of the other hunter-gatherer groups that were living there before. And when you look at this PCA, so this is the West African variation, and when you look at where the gray dots on the geographical map is, you will see they are across Africa. And this is because of the Bantu migrations. So this West African component moved to the rest of sub-equatorial Africa, and today in all of those countries, Bantu speakers are the majority populations. So a very large-scale endemic diffusion, but what we learn more and more about the data that we analyze is that this was actually a complex migration of people. And here we see archaeological data of archaeological sites, and we see these evidences of that there's occupation, and then this was low activity or hiatus, inactivity, and then we see occupation again, indicating possible spread over spread events. So the Bantu expansion possibly was not just one event, but several spread over spread events. And we start to see this in ancient DNA studies. For instance, in this study, we looked at the population structure of Bantu speakers in South Africa, and we see that their genetic variation closely look like the geographic distribution of the different languages, so there's a good correlation. But when we look at where the ancient DNA are grouping in terms of the modern day groups, these ancient DNA individuals were from the same location but different time periods, and they were grouping with different groups. So the younger individuals were grouping with the Nguni groups, and the older individuals were grouping with the Tsonga and Venda groups. So indicating again there might have been different waves of Bantu speakers migrating into Southern Africa. And this will become clearer and clearer as we analyze more ancient DNA data. Just to summarize quickly, so deep human history is complex in Africa. It's probably not a single origin, but maybe it looked like this river network with streams splitting and merging in time. But what we need to do now is to do model testing, to test this isolation by distance versus single origin, versus pulses of admixture models against each other. And there's quite a few studies ongoing that's doing this. So there's exciting results coming from this. But then also the Holocene expansion that we had in Africa were quite complex events. And though we know the sort of broad scales of what happened, we really need ancient DNA to analyze these different fine-scale migrations. Yes, that's all from me. Thank you very much. I just want to thank all the funders and my group. Thank you.
Thank you very much, Karina. That was a fantastic presentation. Super interesting, and see how engineer can contribute to this investigation. Any questions from the audience? No, you don't.
Admixture signals that you find in the African genomes. How are they distributed across the genome? So have people looked if there's like less signal in functional, like well-conserved regions versus not like the Neanderthal case?
Yep. I know at least of this one study on the West Africans that they really try to extract the parts of the genome that I think is due to archaic admixture. And they have done these functional analysis. I can't remember if they got like deserts in the functional, whether it's less functional, but I do know they did identify specific regions which they thought might be archaic introgressions. But I think it's all very uncertain at the moment.
Thank you very much, Karina. So it's time for a break. Let's meet back here at 11 o'clock. Remember to pick up your lunch vouchers from the registration desk. And also, there are going to be staff here from the research infrastructure for the National Genomics Infrastructure, Ancient DNA, and the National Bioinformatics Infrastructure, Sweden. So if you want to know how infrastructure can help you with your research projects, there's a desk next to the registration desk. So our next speaker is Caroline Ahlström Arzini at the Archaeologist, part of the National Historical Museum. Please, Caroline.
Thank you very much. I am an osteologist, so I'm not a geneticist, so I'm going to talk from that perspective. Very, very long time in one sense. DNA have been, we have been working with DNA, but not together with the archaeologists in that sense that we, osteologists, archaeologists, could have the possibility to ask questions. We have to be a part of a big project, and it is the geneticists that ask the questions. So in 2019, when SciLifeLab opened this opportunity for us to participate, I was really happy, and I will tell you why. Osteologists and archaeologists have several questions to the DNA researchers. But one question that I have found as an osteologist that is more interesting to them than others, at least what I have from them, from the archaeologists, is that can you say if people are related, and in what way are they related? So that is what I'm going to talk about today. And we could see that from tracing diseases in families. If we want to, we are interested in a group of individuals that are buried next to each other, and are they closely related? Very often it is a child and an adult. And also if we could use diseases or other things to see if people between several groups, if they are related. And I will start with this one, tracing diseases in families. It started in 1994. I got the question from Per Frölund, an archaeologist. he phoned me and said, I have a small cemetery, but probably belonging to a farm, 7 graves and 8 individuals. Can you tell me if they are related in some way? Could you see could you see if they are related? This is a place called Tierp, north of Uppsala. And then he said, you could look specially at one individual. That individual is very short in stature. And I could see when I got the skeletons that the hips are deformed. I didn't know what it was. I could expect what it was, but I did not know. And since it was part of a cemetery belonging to what he thought was a farm, and he was interested in this, if they are related, he asked me to look at discrete traits on the skeletons. And then I said, you perhaps could see if there is another short-statured individual. And I said, that is because they could be related. And when I got the skeletons, I found another short-statured individual. You see here 1 female and 1 male. They are adults, older than 40 years of age, and they are both around 130 centimeters tall or short. I also found out that they were buried next to each other. So, of course, I was really interested in, could they be closely related? I started with asking medical people what could be the reason to that they are so short. And it turned out that they had a disease called spondyloepiphyseal dysplasia. It's a very rare disease. 1 to 2 children with this disease is born in Sweden today. And it is due to either a mutation or one of the parents is short-statured and have this disease, this dysplasia. And it is autosomal dominant disease. So if one parent have the disease, 50% of the children also get it. And they have several other complications, not only this joint changes. And it is very rare that a couple have 2 children and that they are having this disease due to a mutation. So I expected that it must be father or daughter or mother or son. I started already in 1996 to ask for DNA possibilities. At that time, it would cost me 500,000 crowns to get the DNA analysis. I didn't have that. But then in 2016, I got a phone call from Ashot, Maraghan, and team at the Globe Institute regarding Viking skeletons. And I said, yes, I could provide you with that, but I also want to have 2 individuals from Tierp analyzed to see if they are related in some way. And he did it. And the results, I got it after 20 years, and they are siblings. And that is also very special, since very few individuals with that disease survive to adulthood, and also that you have 2 individuals in the same family. So there must be one parent somewhere, either in this cemetery or in another cemetery. This research also showed another thing, and that is that these siblings had a cousin on Öland, and that cousin was not affected by the disease. And we did strontium analysis of the two individuals that was short-statured from Tierp, and on Helene Wilhelmsson had done it on the one on Öland. And as you see, the strontium analysis for Öland for that individual don't match the baseline on Öland. So that person must be from somewhere else, and you see that it's very similar to those short-statured individual. So that person, that cousin, must have some time in adulthood or adolescence moved to Öland. Now we move further to another group, and that is individuals buried next to each other. This is from Östergötland in the middle of Sweden, Sankta Ingrids convent, and they are buried there around 1000 AD. And it was excavated in 1939 by Berit Wallenberg, an archaeologist. And on these stones there were texts, said that there was a son that called Ygle, and he had a father that has buried him, and that is on one of the stones. And the next stone, it says that Rane, another son, laid the stone over his father, Guve. And of course, Berit Wallenberg was curious about if these skeletons from these individuals were still buried under the stones. So she excavated and found one male under the first stone, but under the second stone she only in the beginning found a skull, and then she found bones scattered. So she thought, okay, this individual is not in its original place. But then she found two other individuals, and then she thought perhaps this individual that is whose grave is disturbed could be Guve. She had an osteological analysis, and the two in the bottom of the grave of Guve was one male and one female, according to that analysis. But then she excavated outside the coffins, outside the stone coffins, and found yet another one. And now she thought maybe this may be Rane, the other son. And then, 1940, she reburied all the skeletons in the place where she found them. And 2011, Richard Hedvall opened the graves once again. And now he called me and said, can you help me, Caroline? Can you see if there is someone that could do a DNA analysis to see if these individuals really are related in the way that the stones tell us? So I first did a new osteological analysis and found out that the wife, the woman, is a male, and Rane is a male, a female. And then I called Magnus Lundgren and said, how much will it cost to get a DNA analysis to see, to find out if these are the real ones that are buried under the stone coffins, and to solve the problem that Berit Wallenberg had, 1939. And yes, it took 80 years to get the answer to that question. And we found out that Ygle, yes, he is the son of Guve. But the female that she thought was Rane is the mother of Ygle and the wife of Guve. So, yes, we solved that. And you should know that stone coffins with skeletons in their original place is very rare. There are 4 cases in Sweden. And we have now given Magnus Lundgren the other two to see if they are in any way related. Then I should talk about finding connections between groups. It is Gotland, and we have 3 groups of stone hunter-gatherers. We have the Ejvide cemetery, and we have the Västerbjärs and Fritorp. And that is from 3200 to 2300 BC they lived. And we wanted to know if these groups have had any contact, married and get families between them. And of course it will cost a lot of money to have all these individuals' DNA tested. So Torbjörn Ahlström, he looked at all these materials and found out that some of them, 4 in Ejvide, 6 in Västerbjärs, and 2 at Fritorp, that have change in their foot that make them flat-footed. And that is 2 diseases, could either be Müller-Weiss disease or Köhler's disease, when you have the collapse of the navicular bone, you see here. And I said, we start with these deceased individuals. Maybe they have some inheritance. I know from modern medicine that there are no gene found for this disease, but you can see that there are individuals in families with this disease. So we started with Ejvide and Fritorp and sent it to Magnus, to SciLifeLab, to see if they are related in some way. And Magnus also compared it to a previous study made by Coutinho and their team, and also got the material from Ejvide included. And what we found was that 2 individuals with flat feet at Västerbjärs, they are related, first degree. But we didn't find a connection between the flat-footed individuals from Västerbjärs and Fritorp, and they have not looked at the Ejvide. But what we have also found is that in Ejvide, they have already found that there is 1 pair of individuals that are related. And at Västerbjärs, there are 3 pairs of individuals that are related, and 1 of them have these flat feet. But it also turned out that is at least 1 pair of individuals that have a connection, biological connection between the group of Ejvide and Västerbjärs. So this is one question that archaeologists are interested in. How many individuals in a cemetery or in different cemeteries are related? This is just the beginning regarding archaeologists asking questions to the DNA researchers. Thank you.
Thank you very much, Caroline, for those fantastic presentation. And we're happy to be able to provide DNA analysis and address these long-standing questions in archaeology. So, any questions for Caroline on her presentation?
It was very clear.
Yes, must have been very clear. Exactly. That's excellent. Yeah.
Presentation. I just wonder, so that was quite a long distance migration between Tierp and Åland. Do you know more about the mobility of people in that time period? Is it unusual for a person to move so far?
we have done a lot of strontium analysis, so we can tell that there are a lot of people moving from one place to another during Viking Age. I have studied people on Gotland, Scania, and the middle of Sweden, and especially places where you have like Sigtuna or early Lund, but also at Gotland, an island, there is quite a lot of people coming from different places. Yeah.
Thank you very much, Caroline. And then our next speaker is Helena Malmström from Uppsala University, who are going to talk about ancient DNA from foragers, farmers, and Neolithic herders in Neolithic Scandinavia. So, please, Helena.
Thank you. Yes, okay, so thank you for inviting me and giving the opportunity to talk about something that I have been interested in for a very, very long time, and that is the Neolithic time period in Scandinavia. Well, one of my interests anyway. So it's actually 10 years ago since we published the first genome-wide data from ancient Scandinavian individuals, but we are many people in this room that have been working and struggling for a long, long time before that to try to get some DNA out of samples, but it wasn't as effective as it is nowadays. So the reasons why we now see this huge boom with publications with hundreds of individuals with lots and lots of data, there are a few different reasons for that. One is the technology that several people has already mentioned. We have the next generation sequencing that enable us to generate large amounts of genome-wide data. It's also that we actually have laboratory methods that fit with this technology and improved methods for DNA extraction to some extent also. And then we know so much more now about preservation in bones also. We know more which regions to target, like tooth roots and temporal bones, the part of the ear bone that is very hard, so DNA is preserved better. But with that said, DNA preservation varies a lot and can vary a lot even within one single bone element. So what I want to do is to try to give you a brief summary of what we have learned this far using archaeogenetic data to study the Neolithic in Scandinavia. This is a very interesting time period. We heard Mehmet talk, for example, about what happened in Anatolia and in the region where domestication started and so on. But to Scandinavia, it took quite a long time before we can see the traces of agriculture and animal husbandry in the archaeological material. It's not until 6,000 years ago that this is something that is shown. Before this, we had hunter-gatherers, Mesolithic hunter-gatherers in the region. But one of the reasons why this is a very interesting time period is because we have different cultural complex. We have different, quite large differences in the archaeological record. And this is just an example. Of this, we have the first farmers in Scandinavia, the funnel beaker culture. Oops, sorry. That was impressive. Okay, so you see the yellow nice stone boulders I've painted there or drawn. It represents the megalithic tombs that these people built and buried many, many individuals together. We find domesticated animal species and so on in settlement sites from this archaeological group. We see tools that are used to farm land, for example, also, and very distinct types of pottery. And then in one of the other groups, sorry, over there, the pitted ware culture is a group that has a livelihood based on hunting, gathering, and fishing, so it's quite different. And they also have different ways of burying their dead. For example, we see fishing and hunting equipment and so on in these burials. And then we also have a third group during this time period, the battle axe culture, that has very distinct burials where objects are placed, or grave goods placed in specific positions. The dead are buried in a crouched position on the side like that. So we do see these differences. This is, of course, a simplified story. There's always more complexity, but we see these general patterns. And questions that is very interesting, at least in my point of view, to ask the archaeological record or the human bones in this case. It's, for example, so if we have many people buried in one of these big stone tombs, for example, and we do analyze them genetically, do they belong to the same population as some other individuals that we have found from some other context or not? It's also very interesting to investigate whether or not these are related in a certain way, and that could tell something about the social structure of the society. So what we investigate is the genetic variation that we see between individuals and between populations. But I also like to highlight the fact that humans have a common origin. We are very similar to each other from a genetic point of view. So what we are talking about here during all of these days, it's those small parts that vary between individuals. It doesn't mean that we are super different from each other, but this variation can give us clues as to the demographic history of individuals and populations, and see if there's been admixture or if populations has like drifted apart and things like that. Okay, so the first farmers in Scandinavia, again, the funnel beaker culture. I don't think I need to introduce that the Neolithization process is the onset of farming practices and so on, because we heard quite a lot about it. But one of the questions that was really interesting to ask the human bones in this case was, so we see these new things in the archaeological record. Does this implicate that there are new people that come with these new traditions, or is it the resident hunter-gatherers that have started to adopt a new lifestyle? So the funnel beaker culture was not only present in Scandinavia, it's present in northern continental Europe also, and they have this quite distinct pottery. Many of these cultures are named after the types of pottery that they have. So this is at least somewhat updated. This is just some example of some of the individuals that we have investigated from this archaeological context. So you have seen a couple of PCA plots previously. What we have on this one is the gray dots, which are present-day populations. And then we have the small color dots that are different prehistoric individuals. And here you can see where our funnel beaker individuals fall, and they are relatively similar to farmers from Anatolia and early farmers from Central Europe also. So they're actually quite distinct from the hunter-gatherers that you can see on a line there that lived in the region previously. This was not expected, as Anders mentioned. The general thought at that point in time was that people had stayed for long times in specific places. We could also see that there has been some admixture with hunter-gatherers, even though the genomic signatures look more like early farmers. But this was quite interesting also. And when we do shotgun sequencing and investigate the genetic data that we get out, we also, as you've heard previously, we get soil bacterial DNA also, for example, but we also can get information about prehistoric pathogens in this data. You have heard some more examples of this. And a study done by the Copenhagen group investigated lots of previously published individuals and looked for pathogens, and they found an early version of the plague in some of our funnel beaker individuals. If we then move just a bit forward in time, we have the pitted ware culture that were these hunter-gatherers that lived in the coastal regions, the purple regions here of mainly Sweden, some in Denmark and southern Norway also, and on the Baltic Sea islands. They have this pit-decorated pottery, hence the pitted ware culture. They on the Baltic Sea islands, they have like big cemeteries, so you have single graves, or you have graves with one or maybe a few individuals in them. So this is very different from the early farmers. Since the preservational conditions are really good on Gotland, for example, this far the published data from this type of archaeological context come from this region, and we have about 25 individuals this far. And if we see where their genetic variation fall, you can see that it's closer to the previous hunter-gatherers, and actually quite far from those early farmers that fell here, even though they do overlap in time, and even though some of these early farmer individuals investigated came from Gotland, also from the Ansarvedolmen on that photo. We have done a few investigations of kinship that is published, not so much like directed towards those questions, but we could see already in 2015, I think, that on the burial site of Ajvide that we have two children that are buried like this. They're quite young, 5, 6, 7, something like that. And when we only had mitochondrial DNA, we could see that they had different mitochondrial DNA, which means that they're not at least closely maternally related. Then combining some of the other data that we had been analyzing, we investigated, or we saw a first-degree kinship between two males on this cemetery, buried not too far from each other, but in two different graves, and they must have been father and son. So if we then move to the third type of archaeological context, we see the battle axe culture. It's was a part of the corded ware complex that is that was quite widespread, and they overlap in time with the pitted ware culture also. And as I mentioned, there's like this formal way of burying people that is quite precise, and this is an example of a burial in Linköping, Bergsgraven. It was also the reconstruction on the first slide I had. And they have these battle axes made of stone, but that are quite distinct. So here also, different individuals from these types of context has been investigated. This is some truth with modification, because actually a few of these individuals have been found in megalithic burials, and it was basically after we got the radiocarbon dates and saw their genetic signature that we saw that they actually fit with this group. But what we can see from these individuals is that they, again, cluster somewhat differently than the other individuals. These have influence from the steppe region. So to try to wrap up the really broad picture of what we know this far, it is that this is admixture analysis. So you have these bars that represent ancestry component. You try to divide the genetic variation within the individuals into different components, and in this case we have the major purple color here representing the Mesolithic hunter-gatherers. If we look at Anatolian farmers in this comparison, they have a very different genetic component, and our first farmers are also similar, but you can see some of these purple in them, which means that they have a bit more admixture than earlier farmers in continental Europe. If we look at our sub-Neolithic hunter-gatherers, the pitted ware culture, you see that it's like reverted. So again, they have more ancestry from these hunter-gatherers, but there's some admixture with farmers. It's less than the other way around, though. And then we have heard a bit about the Yamnaya steppe herders, which are kind of the genetic influence of the corded ware culture. So corded ware culture individuals and these steppe herders have this turquoise component. So you see that something new is coming in, something that wasn't there previously. And that is a component that we also see in this battle axe culture individuals from Scandinavia. So to try to summarize this, we have 3 genetically distinct cultural groups during this part of the Neolithic in Scandinavia. The first farmers were to a large extent immigrants sharing an Anatolian ancestry. The Neolithic hunter-gatherers are more similar to previous hunter-gatherers in the region, which is very interesting because we have a time gap in the archaeological record here. We don't have a continuum from the Mesolithic to the time when the pitted ware culture lived, so this is interesting. And we also have these later farmers and herders that display this steppe ancestry that I mentioned. So quite rough kind of descriptions of the Scandinavian Neolithic, but the question you can always ask is, if these patterns hold with more data, do we see deviations in certain regions and so on? Something that is interesting is that we now have seen in large parts of Europe that these patterns are seen in other areas too, not only here. But there are so many more things we can try to figure out. When did certain of these admixture events happen? Did they happen like in unison, or was it like more of a continuous process, for example? Also connecting mobility information like strontium data with the dietary patterns and burial gifts and so on, and combining all of this together with the archaeogenetic data is, of course, super interesting. And there are so many ongoing studies. So I think that we, in the near future, are going to know so much more about this area. Yes, and this is just, there are so many people involved in all of these studies. These are just a few examples of people and some of the funders that has contributed to this research. Thanks.
Thank you. Thank you, Helena, for that fantastic presentation over this really fascinating time period. Questions for Helena? This also remembered, for those who are online, it's possible to use the Slido tool to answer questions there, and I will pop up on my screen here now. Any questions in the audience?
It was very interesting. I want to ask the Battleaxe genetic profile, does it appear, is it more easily explained as Yamna admixture to was that funnel beaker background or pitted background, or a mixture of both?
We couldn't really discern that completely, since both the funnel beaker culture and the pitted culture are slightly mixed. But I think that it could be with the funnel beaker culture individual, or who knows if there are pitted individuals with slightly more admixture. But I think that also in terms of the types of analysis we can do, we're going to get way further with these questions from now on.
This is maybe both to Helena and Mehmet. I'm wondering about the Anatolian component we have in the Scandinavian material. We call it Anatolian, a little bit sloppy. Is it Anatolian, or is it Levant, or is it Mesopotamian? Are we able to get that detail, any of you two?
Good question, I would say. I think we're struggling more and more. First of all, I mean, since these are studies that are done quite a while ago, before we knew what we know today, things are moving so fast. There's also becoming a problem with nomenclature. What should we call what once we learn more about different cultures? But I think maybe Mehmet could answer a bit more. I don't know.
Best candidate seems to be Anatolia. I mean, Levant and what else did you say? Mesopotamia. Well, Mesopotamia and Anatolia, they were quite close, but and Central and West Anatolians, they seem to be the best candidates.
All right. Thank you very much for those presentations to all the speakers in now these sessions before lunch. So now it's time for lunch. And then we'll meet back here at 20 minutes past 1 o'clock. Feel free to have engaged discussions and browse the displays here. Maybe you can have a look at the mammoths that will be discussed in the afternoon. Thank you. We had some fantastic presentations before lunch, and we continue with very interesting topics, shifting a little bit towards the use of ancient DNA in biodiversity and evolution studies. And first off, we have our second keynote speaker of the day, Pete Heinzman, who we are very happy to have recently moved his research group to the Center for Palaeogenetics here in Stockholm. And he's going to talk about how to reconstruct past animal communities from ancient environmental DNA. So please, Pete.
Hi, yeah, thanks Magnus for the invitation to give this talk. So today I'm going to be basically giving this sedimentary ancient DNA talk to you. Today I'm going to be focusing on animals, mostly vertebrates, but there'll be a bit of invertebrate stuff in there for any of you that like those guys. All right, so let's just start off basic. Like, what actually is ancient environmental DNA? How does it compare to regular ancient DNA? Well, people have already defined ancient DNA, or at least I think they have so far in the symposium. But ancient environmental DNA is ancient DNA that's recovered from basically any kind of substrate, like soil or mud, or any other kind of like an environmental substrate that lacks macroscopic remains, so any kind of fossils. And it's usually originating from a mixture of different taxa, so it makes it for a complex substrate. And why do we use it? Well, if we imagine a scenario where we're in a place and we've found 5 bones, they've been radiocarbon dated, and we want to understand these bones are all from the same taxon, let's say they're from a mammoth. We want to understand when this taxon became extinct. And to do this normally, we would use the most recent radiocarbon date, the youngest dated animal. But of course, you have problems with this approach because, of course, you're missing lots of information and you almost certainly haven't got the last individual, so you don't really know when the animal went extinct. With sediments, however, you can focus on very short, fine-scale time intervals, which you can't do with bones, and you can be very targeted in how you approach that. And then you can test for DNA within those sediments, and you can understand when that animal went extinct. And not only do you see when the animal was present, you can also try and detect absences too. Okay, and the real strength of ancient environmental DNA compared to other approaches that are commonly used in paleoecology for understanding when and where things were, is that it has the trinity of taxonomic breadth, i.e. you can detect multiple taxonomic groups. It's got high taxonomic precision, which of course it shares with regular ancient DNA, and you have high temporal resolution. And this is why sedimentary ancient DNA is so good for paleoecological reconstruction, which is what the focus of my talk today will be about. So where do we get it from? We can get it from a number of different sediment types, caves, permafrost, archaeological sites, and other open sites. And then we can get them from closed catchments, such as from lake sediments, and also from more kind of like hard-to-reach areas, such as the sea floor. And the things that these kind of like substrates have all in common is that they provide a really nice record of biodiversity living in the local area. For things like permafrost, and you get DNA from quite a large area, whereas lake sediments, it's more catchment specific, and then caves, it's very specific to that area. And you get a nice steady accumulation of the sediment through time. And this provides very nice fine temporal resolution, which, as I said, was the key to why this substrate is so strong. And with this, we can reconstruct, when we sample these sediments through time, we reconstruct ecological communities from across quaternary landscapes. And just talking about ancient DNA more generally, we have these various different sources of ancient DNA, which I think we've, at this point, covered throughout this symposium today. You've got tissues through to things like microbiomes, and then through to various different sediment types. And as you go across these, what you're doing is you're increasing the area that your DNA is from, and as a consequence of that, you're increasing the complexity of the mixture that is present. So it becomes harder and harder to analyze. So how do we actually go about doing this? How do we recover this ancient environmental DNA? Well, one of the main approaches is basically the shotgun approach. Most of you who work with ancient DNA are familiar with this. This is the kind of like go-to approach when you're working with bones, where you simply randomly sequence anything and everything that is in your DNA extract. But there are also alternative approaches that are targeted, either using PCR-based approaches, which are super sensitive, or target enrichment approaches, which have the advantage of retaining DNA damage signals. And then once we have our data, regardless of which of these approaches you use, you go through, you need to authenticate your data and you need to identify your data. And this is where the importance of reference databases is super important. If you don't have good reference databases, you can't identify your data. Okay, so that's the background to all of this. Let's go into a nice story about single species reconstruction. And for this, I'm going to talk to you about the St. Paul mammoth. So, St. Paul mammoth. So St. Paul is an island off the coast of Alaska, and it hosted one of the last woolly mammoth populations to have ever lived. So we all know Wrangel Island. This is where mammoths survived until the latest, around 4,000 years ago. But mammoths actually survived on St. Paul until around 6,500 years ago. You can't see it here. It's only about 2 pixels big because it is a tiny, tiny island. Really tiny. It's amazing that mammoths were surviving here for thousands of years after they went extinct elsewhere. So how do we know when mammoths became extinct on St. Paul? Radiocarbon dating of bones. So as I introduced earlier, you can radiocarbon date bones to work out when things go extinct. This is a small island. There aren't many mammoth remains, so we didn't really know when they went extinct with high precision. But mammoths are elephants. Elephants drink a lot. They visit lakes. So we should get a lot of mammoth DNA in lake sediments. And very handily enough, on the center of St. Paul, there's this really nice lake record, which is one of the only freshwater sources on the island. And we know from previous studies it's got a sediment record stretching back around 18,000 years. So it's the perfect study system to examine when and why mammoths went extinct on this island. So our colleagues went out, they cored this lake in the wintertime. Then we sampled the sediments for DNA, and we shotgun sequenced them. So we generated basically data from anything and everything that was in the DNA extract. So when you do this, how do you actually identify mammoth from all this other kind of like ecosystem DNA, all this microbial, all this plant DNA, DNA from other animals, etc.? Well, what we can do is we can start by just simply aligning to genomes. So in this study, we aligned to the elephant and the woolly mammoth genome, which was not quite publicly available yet. We also aligned to the two-toed sloth genome. And the reason to do this was to kind of understand what our kind of like randomized mapping rate was. Two-toed sloths, they're endemic to South America. Don't get sloths up in the Arctic. At least you don't now. You did during the last interglacial. You had giant ground sloths. But during this time period, around 6,500 years ago, no sloths in the Arctic. So this is a complete kind of like control. And then to kind of like remove any other crap that basically may have mapped to this, we removed anything that also aligned to the human genome, to the horse and cow and mouse genome, and any other kind of like things that could be identified as microbes. So once we'd done all that, the first thing we wanted to do was try and authenticate this, examine DNA damage patterns as shown here. And what we found is that the reads that were aligned to the mammoth and to the elephant showed really nice fragment length distributions and really nice deamination profiles. Conversely, the reads that aligned to the two-toed sloth didn't look like this at all. This was all random mapping, basically, of really short ancient DNA reads. And the take-home from this is when you're dealing with sedimentary ancient DNA data, if you have very little aligning and it's all very short and it's not showing anything useful, no damage patterns, it's almost certainly a false positive. And this was a really nice way of showing that we actually do have mammoth DNA versus kind of the random crap. Okay, so that's showing that there is mammoth DNA in this record, but how did the distribution look through the record? So what we have here on the x-axis is time from 12,000 to present, and then on the y-axis we have basically the amount of DNA that's aligned on a log scale. This is, for each of these samples, we have the amount of two-toed sloth DNA, so this is basically our baseline. When we add on the mammoth DNA, you can see we get this sudden decrease at around 5,000 years ago. And this is really cool because in basically every sample prior to this, we get tons of mammoth DNA. And then as soon as we get past this interval, all the samples show this basically the same amount of mammoth DNA as they did the two-toed sloth. So this is where we infer that the mammoth has disappeared. And this was actually around 5,600 years ago. So this has basically brought forward the extinction timing by around 900 years. And what's really cool about this is this, as I said, this record goes back around 18,000 years. It's 13 meters, and we isolated the time, the distance in the core where this interval to 2 centimeters. So this is really showing the high precision that you get from these. And this was basically, this result was confirmed by looking at additional paleoecological proxies in the profile. So here we've got fungal spores, which are characteristic of large herbivores on the landscape, and these disappear at basically the same interval as the mammoth DNA. So this was really nice verification that this is what was actually happening. And why was this happening? Well, we can look at other paleoecological and paleo-environmental proxies that are preserved in the lake sediment. And what we found is that there were an increase in these kind of like more salt-tolerant cladocerans. And this was suggesting that actually the lake was becoming more and more salty. And so the suggestion is that these mammoth died because of local freshwater availability issues. So that's kind of like a single species reconstruction, but I'm here to talk to you mostly about animal community reconstructions. And there are a number of these that have been published from different substrates, so I'll just shoot through a few of them. So this was one that was published by Elena Zavala and colleagues from Leipzig, and they basically reconstructed the mammalian community reconstruction through 300,000 years at Denisova Cave. And they found really nice turnovers of different elephantids, hyenas, and bears at different interglacial and glacial periods. Just kind of going to another part of the world, in the Yukon territory of Canada, which is right next to Alaska, another group, in Hendrik Poinar's group, they reconstructed a steppe environment from permafrost and showed basically that during the last ice age, on the left-hand side of the plot, you have all the kind of megafauna, and then they disappear as you and transition into the warmer period of the Holocene. But what's interesting from these kind of studies, and this is some real take-home for when you're dealing with open sediment sources, especially permafrost, is they're also finding that you get really late survival of megafauna. So on the mainland in North America, horse and mammoth are thought to have gone extinct around 10 to 12,000 years ago, but they were finding mammoth and horse DNA in very low amounts until around 4,000 or 5,000 years ago. This is extremely controversial because there are no bones that have been dated to this age, and it could well be that there are taphonomic issues at play, although this is obviously an area of further research. And then moving on to lakes, a really nice study was showing that the community reconstruction in the ice-free corridor of Canada. So what's happening here is, at the end of the last ice age, we have 2 ice sheets covering Canada. And then basically, at the end of the last ice age, as they begin melting, these 2 ice sheets kind of unzip from one another, and they open up this ice-free corridor. And this was important because it was thought to be a colonization route of the Americas at the end of the last ice age. But this study, along with another one, showed that basically this didn't open up until around 12,600 years ago, or at least became biologically viable. And this is much later than the first peoples that are known from the lower part of North America. And this is basically some of the prime evidence that people probably used the Pacific coastal route as the way to get into the more southerly latitudes of North America and South America at the end of the last ice age. Okay. But we can go beyond all those examples I showed. They were basically mostly mammals, maybe a few birds, but we can go further than that. We can look at other animal communities. And I'm going to show you a nice example that we just published, a 13,000-year-old record from northern Norway. Now, this is a lake sediment record, and it's interesting because, one, it's close to an archaeological site, so we wanted to reconstruct that, but also it's right on the coast. And as you can see from the map in the center here, you've got these kind of like raised beds. And what these are, this is from when this 13,000 years ago, this lake was right at sea level. It was right on the beach. And then as the ice sheet has melted on the Scandinavian peninsula, you have isostatic rebound, so the land has raised. And these are basically raised beaches since the end of the last ice age. And we reconstructed a full faunal record through time from this lake. And we got not just mammals and birds, but also various invertebrates. And what was super nice about this is that we saw this really nice transition from this marine beach around 13,000 years ago to when the beach basically got isolated. We no longer really get any of these marine taxa, and we start getting these terrestrial taxa. We have some very common ones like frog and various large mammals, which are kind of like characteristic of northern Fennoscandia today. But then during the middle Holocene, so this was a period that was slightly warmer than it is today, we actually get the occurrence of more forest-adapted taxa like beaver. So up there today, it's near the tundra, near the tree line, just a few birch trees, but not a huge amount. But here it's showing that there was a lot more birch and probably some of the trees too. And what's really cool about this is that nearby archaeological site that I mentioned, they also have beaver bones that have been directly dated to the same age as this detection in the lake sediments. And then what's very interesting from an archaeological perspective is we also get this really nice reindeer record, especially towards the top, where we see a kind of like gap in the record, which corresponds to the Sami medieval period, where we don't think that there was much reindeer grazing in the catchment. But then during the Sami Iron Age, later on, we get loads of reindeer DNA. And this is suggesting that there was a lot of probably heavy grazing within the catchment. And we got a weird result here. So you may notice that there's like no terrestrial taxa where we have the marine taxa in the early part of the core, but then we get this kind of like lingering signal of marine kind of like taxa, and it's basically pretty much just whales. And we're trying to work out what this was. Is this cross-contamination? Is it errors in the databases? All this kind of things came to mind. It turned out we couldn't resolve it using any kind of like technical error, so kind of came up with the hypothesis that this might actually be to do with sea spray. This works because in northern Norway, the environment is quite cold, it's quite damp, there's a lot of fog anyway, so you can expect preservation of sea spray basically drifting just inland from near the ocean. Is this right? Is it complete and utter crap? I don't know. We will find out. But we've put the hypothesis out there, happy for people to test it. All right, so that was from kind of like Arctic Norway, where it's kind of like a very natural environment. But of course, we also have archaeological records and or catchments that have got a lot of human impact. And this is very typical of the Alps. So we just published this paper in Nature Communications just earlier this month on a lake record from Switzerland. And what we've done here is reconstructed the local environment, not just using DNA, but a suite of other proxies to understand how the impacts of the joint impacts of climate and humans through time. What we find is in the first part of the record, from around 12,000 to 6,000 years ago, it's a very natural environment. We only really find wild animal taxa and very low incidences of other proxies of human impact. Ignore this kind of one detection here of cow. The problem is with cow is it's a common contaminant, and we always just get sporadic kind of like occurrences of it. So when you see these kind of plots, especially for domesticate taxa, and you just get a single like detection like that, you always treat it with a bit of caution. And this is a really nice example of that. Then we move into the late Neolithic. We don't detect any kind of like domesticate taxa, so the only real taxa we're detecting here in the animal data is red deer. But we do have pastoral indicators, and this is from the pollen and from the sedimentary plant DNA. So this is suggesting that people are farming in the area, but not with animals. However, later on, from the Bronze Age onwards, we start seeing the domesticates coming in, and this corresponds really nicely with the pastoral indicators that you see in the floral taxa. We see sheep coming in first, and then cattle and goats coming in much later, probably during the Roman period, but certainly by the Middle Ages. So where's all this going next? So I've basically presented to you today a load of studies from single sites, but really this area is moving into kind of like multi-site contexts. And this is really important because you can have all sorts of biases in a per catchment area. But if you combine lots of catchments from a whole region, you can start doing regional scale interpretations. And people have started doing this with plant records now, but we're moving into doing this with animal records too. And a group I used to work with and I'm still collaborating with, we're doing this from northern Norway and from the European Alps, but we're hoping to move into other regions too. And the other key thing we want to do is go much further back in time. So most of these are focused on the Holocene, but we know that now that sedimentary ancient DNA and ancient DNA in general preserves for hundreds of thousands of years. So there's a lot of potential there to reconstruct major changes through multiple glacial interglacial cycles. And then there's environmental paleogenomics. This is kind of like a really new area, and this goes beyond just reconstructing communities, where we actually get population genomic data directly from the sediment. Sedimentary ancient DNA, it actually provides a population sample, so you can't understand individuals from it, but you can understand populations. And this means you can do things like genome-wide analyses, like PCA and admixture and phylogeny. These are some things that we've already seen today in some of the other talks. And you can look at allelic presence and absence. What you can't do, and this is what you can do with single genomes, is things like phasing or any analyses that require heterozygous sites like PSMC, runs of homozygosity, and allele frequency distributions. But what have we managed to be able to do with this? So one thing is, this is from a cave site in Mexico, where basically some collaborators, they were looking for humans in these Mexican cave sediments. They didn't find any humans, but they did find bear DNA, and this bear looked really weird. And then they put it in, and they figured out that actually it was a mixture of two bear taxa in the single sediment sample. And they were able to actually pull out these two bear taxa. We have giant short-faced bear. This is extinct in North America now, and or globally, sorry, and the black bear as well. Now, the giant short-faced bear, we don't actually have any genomic data for. We actually had to generate a reference genome to be able to identify the DNA within that study. So we couldn't actually do anything really interesting, and this really highlights the value of having reference databases. The black bear, on the other hand, has lots of reference data, and because of that, we were able to basically place it into a population level analyses and showed that the Mexican black bear, the Mexican black bear is actually more closely related to the eastern black bear of North America. Another study that was published at around the same time, and this is amazing because this is from just one single sediment sample, less than 0.5 gram of sediment, and they managed to pull out genome-wide data from a wolf, a bison, and a human. And because all three of those taxa have large comparative reference data sets, they were able to basically understand which population these three taxa were from. Because it was just one sediment sample, they couldn't really do anything cool through time, but they could verify that the wolf and bison and human haplotypes and populations that they got were known from the area. And this was especially well done for the humans because the sediment sample in the admixture plot is shown right here. And right next to it, this is a skeletal individual that was found from a nearby cave dated to around the same age. So it's basically showing that, yeah, you can place this sediment data into known population data that we have from bones, and it makes sense. So that's good. So just in summary for this, we can now recover most components of vertebrate communities, and although the methods are improving, the records are still scattered, and that's because these methods really still need quite a lot of development. But there are many emerging methods, and we're only exploiting usually a tiny amount of this sedimentary ancient DNA. That might be because of the methods we're using, or it could be also because the reference databases just don't exist for identifying a lot of it at the moment. And the boundaries are still being tested. We don't know how far back in time we can go with sedimentary ancient DNA. Can we go back more than 1 million years? Can we not? And also which environments we can get sedimentary ancient DNA from. And because of this, there are still many questions that are left to address. Cool. And I just acknowledge my kind of like previous research groups and my funders. Thank you.
Thank you very much, Pete. That was a fantastic overview of a super interesting and fairly new application of ancient DNA research in sort of ecological reconstructions. So questions for Pete then?
Nikolai?
Super interesting. Thank you very much. You emphasize the temporal resolution of ancient environmental DNAs, which is of course great advantage. It's super attractive. But I wonder, so the dating of ancient environmental DNA is based on probably the level where it was found, the layer in soil or sediment. So how confident is this? How confident is the dating of ancient environmental DNA?
So for things like lakes, this can actually be the weakness. So in the example I gave you for the last mammoth, where we were down to 2 centimeters and we got super high resolution within the stratigraphy, what we were most limited by was the age depth model. And the way we construct the age depth models is we don't necessarily have a radiocarbon date from exactly the same sample that we took the DNA sediment from. We might have various radiocarbon dates throughout the sediment core, and then we could basically construct a Bayesian model reconstructing the relationship between age and depth. And that is not always linear. It can, depending on sedimentation and things, can be a little all over the place. And that is can be the limitation, yeah, for sure, on the dating. For some things, if you have something like a tephra or something that's really well anchored in your core, be that permafrost or be it a lake, that can provide a really nice temporal resolution. And yeah, there are various other dating methods, but it can be problematic, especially if you've got any reworking. And reworking is definitely the Achilles heel for this.
This one, okay. Thank you, Pete. Excellent. And also, by the way, really happy to have you at CPG now. Excellent. Question here, something I've been curious about. How have you done this on a lake where there is pollen as well? Do pollen and DNA overlap, or do they show completely different things, or do you have an idea?
Yeah, so they are usually in a lot of agreement, but the problem is they're not always directly comparable. So for pollen, you can usually identify a lot of woody plants very well, especially trees. The problem is with tree pollen is it's usually long distance dispersed and it's not local to the catchment. And DNA is really great because it is local to the catchment. So you can sometimes see a little bit of discrepancy there. But for a lot of the other interesting plants, which are basically the flowers, the herbs, and the grasses, pollen is not so good, especially for the herbs, at distinguishing those taxa, whereas the DNA really is. And that's where the DNA really complements any pollen diagrams by really highlighting the diversity of the herbs.
I would like to remind the online attendees also that you can post questions through the Slido app.
So I have kind of a silly question. Have you seen any whale hunting in the lake in Norway? That some kind of human post-processing of remains close to the water, at least to me, it seems to more likely explanation.
Yeah, no, we thought about that. Another explanation actually was that you had erosion of these kind of like older layers from the side of the lake, kind of because there's a scree slope nearby, which could also explain it. But the problem is right now, so those whale detections, they're quite late in the record, around 4,000 to 2,000 years ago. And by that point, the lake level was already around 80 meters above the sea level. And by that point, you probably wouldn't expect people to be taking any whale remains that far up. So, yeah.
Thank you for the talk. It was really inspiring. My question is, how do you date the DNA samples you take from, for example, lake to, like, for example, 4,000 years? How can you say that it's from 4,000 years ago, like 6,000 years ago?
So as I said to Nikolai's question, we construct these age depth models from lakes. So we will take, so if we got like say a 2-meter core, which was the case in this lake, we sample say 10 to 20 little macro remains of plants. So a bit of leaf or a seed or something like that. Then we can radiocarbon date those at specific layers. So we then have a date per layers, and then we build a model between those dates, and this is called the age depth model, and we basically infer the age of the sediment DNA sample from that age depth model. And this has been commonly applied through for paleoecology for decades. Carl.
The people online won't hear you, no matter how hard.
Okay, maybe more common that what with the pollen and the DNA. Yep. That what we, so, well, I'm a geologist, so I didn't do this, but collaborators doing ancient DNA. What we found in the Urals was that, as you said, you get the far-traveled pollen. But that's also a good thing, because they complement each other, as you said, with the going down to species level in some instances. But also that then from the pollen, you can see something is nearby, but not in the catchment. So that's also a big benefit of using both and comparing them. So you can get more of a regional signal from the pollen and more a local signal from the DNA in the best case.
Absolutely. It depends on you, completely on your question. If your question is catchment specific, then the DNA provides a much higher resolution. But no, but the problem is with some long distance dispersed taxa is it can be really long distance, like thousands of kilometers, right? But yeah, but this is one of the reasons why we want to kind of like move these fields into more regional studies, because we always get catchment specific kind of like oddities with these things. Like, for example, in the northern Norway examples, we have one catchment, we have the catchment where it was a beach at the beginning, and then we have another catchment where it's glacially fed, and this kind of like has a bit of a control on what you find. And this is why it's important to get multiple sites to kind of build up a full picture, because of these oddities that occur at a very catchment specific level.
Okay. So it's a question about DNA moving in the environment through sort of, water movement or diffusion. How do you deal with that potential?
Yeah, so this was a well, it still is an issue, but what we found, and this is from comparing the plant DNA to pollen and things like this, when the pollen doesn't move because pollens are macrofossils. But we find actually very tight agreement suggesting that in waterlogged sediments at least, you don't actually get this vertical movement of DNA. We also found that in the mammoth where we had a really nice agreement with the spores. It's like we didn't have any evidence of diffusion of the mammoth DNA into other layers. So you have that kind of verification. Now, this is true in waterlogged sediments and most certainly in permafrozen sediments as well. But in dry sediments, such as caves and kind of like open sites, the vertical movement of DNA is a real problem. Especially in open sites where you might have biotubation from root penetration and active layers as well. And this can cause major problems. So yeah, if you're taking surface archaeological sediments, be very aware that this can be a problem. This is why it's important to look at DNA damage and just check that there's intact stratigraphy at a minimum, but you could still have issues with water percolation.
Thanks. Yeah. Further question? I think there was a mic somewhere. Yeah, that's it, please. It's coming. It's coming. Yeah.
Okay. For the lake in northern Norway, you show human DNA as old as 12,000.
Yes, so that whole red bar, those are our assumed contaminants. And this was because that particular record was generated with PCR-based metabarcoding. And in that particular method, you cannot distinguish modern from ancient DNA. So, and that is a huge problem with human, because whenever you want to report anything as ancient human, what you have to do is to be able to demonstrate that there are deamination profiles, and you lose that from the PCR. So in that particular plot, I had those taxa in red, and those are the ones that we have as assumed contaminants, including human.
But still, if you, there is very close by, there is archaeological evidence that the humans were present in the ice marginal environment in Varangen Fjord. I don't know if you are aware of that.
Absolutely. The Morten's Nest site, where so that lake is right next to the Morten's Nest site.
So then
it could be, yeah, absolutely, but the problem is with this is we always get human contamination in these experiments because humans are mammals and these primers are targeting mammals. So, and
if you can back it up with archaeological evidence, you would say it's strong enough.
But we also see human in the negative controls, and that's the problem.
So can you reduce this contamination somehow in the future?
So we use human blocking primers, which try to reduce the amount of human amplification. But the problem is if you have very little template, which is very typical in these kind of DNA mixtures, they don't always work 100%. You'll always get some coming through. So, yeah, no, if you want to try and get ancient human from these kind of sediments, you have to use alternative approaches, which will capture the deamination profiles. So it's not to say this will never happen, but it just won't happen with PCR metabarcoding.
Yeah.
Right, last question.
Do you hear me?
Yeah.
Good. Thank you for an excellent talk. I was wondering, do you have any plans to try and test this sea spray through stable isotopes for, say, sulfur?
No, I had not thought of that. That's a good idea, actually. So in the paper, we posit ways of testing this as actually to look at relative deamination profiles. Because if these are basically the difference between the two hypotheses that we posited, which was basically erosion of like older sediments into the lake, and basically this is basically older DNA that's getting redeposited versus kind of like fresh DNA that's getting blown in through the atmosphere, we should see differences in deamination profiles if we did shotgun sequencing, but we haven't done that yet. So that was our kind of like idea of testing it, but yeah, stable isotopes is a good idea. Yeah.
All right. Thank you very much, Pete, for that excellent presentation and for that really also rewarding discussion afterwards. I think that was also very illuminating. So thank you very much. So I'm actually going to introduce myself now. As I mentioned, I'm head of the Ancient DNA unit at SciLifeLab. So this is a talk about what sort of support as a service there is for analysis of ancient DNA for research community in Sweden. So I'm going to give an introductory presentation. We're also going to get a presentation from Tom Martin, who is a project coordinator at the National Genomics Infrastructure. So of course, as you've seen today, and as you will see during the day, there's many reasons why to study ancient DNA. Basically, whenever there is an interest in sort of the temporal aspect of genetics, how was things in the past, how have it influenced the present, and how can you use it to extrapolate into the future? Looking at human evolution, admixture, ecology, evolution, human migration, development of agriculture, structure of prehistoric societies, history of pathogens, and of course analyzing actual phenotypic traits to the extent it's possible. But there are many challenges to working with ancient DNA, making this technology not easy to access directly, to set up your own labs and your own routines to do so. It has been becoming cheaper with new technology, but still that's put it outside of the realm of possibilities for many researchers, at least until recently. The problem why you need this sort of special procedures, of course, is that DNA is a fragile molecule. It does degrade over time. It becomes shorter and shorter. How fast that is, it depends on environmental conditions. DNA also gets modified over time, so the actual information in the sequence does change. That is, as we have heard, is actually a blessing in disguise, because we can then go to the DNA and actually look at it. Does it have this sort of telltale patterns or chemical modification, which is an indication of it being bona fide ancient. Another problem is, of course, the presence of non-endogenous DNA in the sample, so that's not a contamination, it's part of how the sample is like. And another severe issue, of course, is actual contamination of DNA, ancient DNA, with modern DNA in the lab, which either sort of precludes valuable analysis or makes you misunderstand what analysis actually is being, or what data you actually have. So the way to work with ancient DNA is, of course, to have know-how and experience in selecting material that has a high likelihood of actually containing DNA. So select quality material to sort of, just spend your resources wisely. Use protective equipment for the staff to avoid contamination. Decontaminate the material to sort of reduce the amount of non-endogenous DNA. To have clean rooms for the lab, with positive pressure, so you keep, contemporary dust out. Have airlocks for accessing the labs, not moving between labs and transferring DNA. Having specialized laboratory procedures adapted to this type of material. And of course, validation of ancient DNA, coming back to this chemical modification I talked to earlier. So it is quite a setup to actually do this. We are very fortunate to have very strong research environments in Sweden with labs for this. And historically, lots of research has been done either in these groups or through collaborators with these groups. But with interest growing, it's a limitation of access through research collaboration. And that is why there is an increased focus on infrastructure support for ancient DNA research. So where there is service laboratories that do not engage in projects in order to have publications themselves. They don't have specific research questions, but they're just interested in supporting other researchers. So in Sweden, we have mainly 3 organizations supporting this. We have the Ancient DNA unit, which I'm head of. We have the National Genomics Infrastructure, and we have the National Bioinformatics Infrastructure Sweden. So the Ancient DNA unit, we have specialized laboratories. We can help with sampling and so on. NGI does various types of analysis of RNA and protein, also some data analysis, and at large scale. And National Bioinformatics Infrastructure Sweden, they can provide specialized data analysis for these types of questions. But then, of course, all these three infrastructures is part of SciLifeLab. You see our poster signs here everywhere. Many of you are sort of aware of it, using it, but what is actually SciLifeLab? It's a direct government initiative to support life sciences. It has a wide range of different types of units and services. I think more than 40 units currently, 3 of which I mentioned in the last slide. So it's a government-appointed mission to provide research infrastructure in this field. And it's highly distributed. You can find probably various types of SciLifeLab units at all major universities in Sweden, specializing on basically depending on what knowledge there is at that particular given research environment. But the Ancient DNA unit is based at these strong Ancient DNA research environments here in Sweden, at the Department of Organismal Biology and at the Center for Palaeogenetics here in Stockholm. The Ancient DNA unit is also part of a wider infrastructure. We're part of the Hyperion Heritage Science, which is an EU-funded infrastructure, and the Swedish Digital Archaeology Infrastructure, which is a VR infrastructure to support data management and data handling of archaeological data, including ancient DNA data. So what can we actually do from the perspective of Ancient DNA unit? So of course, an important part is engaging in project planning, assisting with a discussion about how can an ancient DNA analysis support whatever research a researcher is engaging in. We can help with sampling in clean room, potentially also on-site. We can perform DNA extraction from various types of material in this clean room environment. We can process this DNA to make it suitable for DNA sequencing. We can check those DNA libraries before actually sending them for sequencing. And then we can assist with DNA data production. But we actually don't do it ourselves. We have an excellent facility, which Tom Martin will talk about, the National Genomics Infrastructure, who actually does the DNA sequencing. We can then retrieve the data from NGI, and then we can do various types of sort of standardized analysis. For example, the ancient DNA content, sex chromosome, maternal and paternal ancestry. We can look at close kinship. We can look into microorganisms in the sample, and to some degrees also custom analysis, depending on our expertise and capacity. But also here, it was the bioinformatics platform, the National Bioinformatics Infrastructure Sweden, comes in and have a much greater capacity and experience and can provide a lot more support for data analysis. A little bit on ongoing development. This is a fast-moving field, ancient DNA research. So as I mentioned, we're looking into making data available for researchers in a bigger context through digital archaeology infrastructure. We have development projects for increasing our knowledge and ability to support analysis of ancient plant DNA, further developing microbial and pathogen analysis, and also looking into sediment analysis. We think is a really major field in the future when it comes to ancient DNA. So with that, I'll leave the word to Tom, who's going to talk a little bit about the infrastructure for DNA, RNA, and to some degree also protein analysis.
Okay, thank you, Magnus. So we've heard some fantastic talks today about ancient DNA and DNA sequence. But how do you actually get that DNA sequence? That's where we can help you. So I'm from NGI, which is the National Genomics Infrastructure, and we provide access to technology for large-scale genome sequencing and genotyping, and we also provide bioinformatics support. So we provide access to this large genome sequencing through our large technology platforms that we have. We have a huge DNA sequencing capabilities for both long read and short read. I guess short read is a bit more your thing in this meeting. We have 5 NovaSeq platforms at the moment. We're expanding constantly. So we will soon be acquiring the NovaSeq X, which is the new system. So we are producing more and more sequencing data at a lower cost for you. And NGI is based in Uppsala and Stockholm. So we're 2 nodes. So you can contact us at NGI, and then we can decide, or we can help you find which node will be able to help you best. So we can direct you to Stockholm or Uppsala. So how does that work? Well, first of all, when you get in contact with us, you can fix a meeting if needed. So we can talk to you and try and understand your question. And we can also involve NBIS, which Magnus talked a little bit about, and we'll talk more about after my talk. The next step is you have your DNA, and we need to get it into the sequencer. Now, we can't just put it straight into the sequencer. We need to make libraries from it, and we basically stick little adapters onto your DNA, and then we can put it into our sequencing machines. You can do that yourselves, and you can give us your libraries. You can talk to Magnus at the Ancient DNA facility. They can help you with all that. Or if you want to, you can give your DNA directly to us, and we can make the libraries for you. The next step is the sequencing. And this is where our fantastic sequencing platforms that we have can help you. So we sequence your data there at NGI. When your sequence data is done, we will run a QC on it, and our bioinformaticians will do all the demultiplexing and everything so you get your sequences back. And this will be a good time to talk to either Ancient DNA or NBIS to help you to decide what you actually need to do with your data. So how do you get in contact with us? Well, we have a website there, and you also have an email. So you can drop us a line, ask us any questions. We're here to help you. I'll just put a nice little picture of some of the things we sequenced. Yeah. We also have a little stand outside, so come and talk to us. We'd love to hear your questions and interesting projects.
Thank you, Tom. So then I just finish off with a little bit information about the National Bioinformatics Infrastructure Sweden. So this is also a large operation similar to NGI with lots of capacity, a very wide diversity of experience. So they can provide support with data analysis in various ways. They have sort of short, free discussions on your project, no sort of hands-on work. But then they also have this fee for service where you get a highly discounted analysis, but you pay by the hour. And you basically apply for a number of hours, and then you're approved. The other option, I have long-term support. So this is a review-based project. So then you apply for resource, and a few projects are selected, and then you get a lot of support, typically 500 hours over 1 to 2 years. So that, of course, is a massive amount of support. It's very valuable. It's also very important that you connect with the NBIS experts, so you actually transfer knowledge to your research team or yourself for doing this analysis. So it's not so just performing analysis, but actually transferring the knowledge how to do this. And also the option of partner projects where you can sort of hire a bioinformatician to work in your team under various conditions. So please, you can reach out to NBIS for discussion these different options, what they mean and what sort of projects they can support. There's also these drop-in sessions that NBIS arranges, very valuable, so you can meet them, and just discuss whatever projects you are interested in doing and getting, hands-on support with various types of solutions to your analysis needs. So with that, I would just like to thank for the time to talk about how we can support research. And as Tom mentioned, we're going to be outside in the coffee break. So maybe the easiest way to get in touch with us is just to come and talk to us. Otherwise, find us online, send us an email. We are happy to help. Thank you. Right. Any questions to me or Tom or NBIS for that matter? We have the experts here in the room now.
Hello, just a quick comment. The workflow that Zoe presented this morning we used to identify microbes, that was done through one of these long-term supports.
Yes, absolutely. Yeah. And we're also using that. So it's rings on water. Further questions? I don't see any questions online. Anything else at this point? Yeah. Sorry. Wait for the mic. So everybody hears, also the people online.
So I have one comment from the NB site. We are really happy to actually be involved as early as possible in the project planning and study design phase. So please don't really hesitate to contact us early in your studies.
Right, exactly. Yes, that's a very good comment. So of course we can engage in that also together, project discussion together with NBIS. Starting off on the last session here now, we are very happy to have Mike Martin here to give a presentation. Mike is going to talk about herbarium, evolutionary holo-genomics using herbarium specimens. So please, Mike, go ahead.
All right, well, welcome back to the last session. Thanks to all of you for coming to see. Thanks to the organizers for the invitation. I thought that was really nice. My initial thought was to, since it's an ancient DNA conference, to present on some other more ancient lines of research that we do in my lab, like on ancient reindeer, etc. But I think I'm sort of being asked to provide a dose of diversity to the talk, so I'm going to talk to you about plants, and I think it's the only talk about plants. So those of you who like plants should get ready for that. So there are a lot of herbarium collections out there. There's a map of around 3,000 herbarium collections around the world. All of these collections contain very many voucher specimens from plants that were often quickly dried after collection by botanists and then placed between these two paper sheets and then archived sort of like a bookshelf situation, like books on a bookshelf in the herbarium collections. So they're overall, these collections are really nice museum resources for taxonomy and morphology and environmental studies, et cetera, et cetera. And they're really well meticulously curated with a lot of metadata associated and usually in pretty good condition. And the number of these, the number of collections in these global herbarium really increased quite a lot from the year 1800. But even before that, you know, the earliest herbarium collections were started sometime in the 1500s. So there are some historical specimens going way back into the sort of golden age of exploration. And as I mentioned, these collections have been used for all sorts of taxonomic, and there's been quite a lot of uses that are increasing all the time for the herbarium specimens. But what I would like to suggest is that these are actually really nice genomic resources, particularly because of how well preserved the plant collections are, and the abundance, and how well organized the collections are. They're particularly open for the sort of population genomic studies. So I thought I would start by just reviewing a little bit about what's been done for historic herbarium specimen genomics. You would not be surprised, I think, to know that actually quite a lot of work has been done on these collections going back 10 to 12 years. First of all, when I go through this quick review of the field, you will see that I'm basically tracking the career of Hernan Burbano at University College London, who has done a lot of pioneering work in the area, he and his group. And so you'll see his name up here. The initial work by his group was basically showing that, demonstrating that the DNA is well preserved in these specimens. It decays rather quickly, so they become short 6 times faster than bone. The decay rate of DNA in these plant specimens. But the endogenous content is often very high, and I think this is because the specimens are so quickly dried after they are collected. So that's nice. And DNA damage does accumulate in a sort of clockwork pattern, and so we have seen this before. There's not a lot of damage, but it is there, and it increases as the specimens get up to 200, 300, 400 years old. Some of the initial big works in the field have been tracking the movement of plants and historical period. For example, this work talked about the movement of potatoes. The work on the left is about the movement of potatoes from the New World into Europe, and the selection on the genes that control the day length adaptation in the potatoes, and also the introduction of Arabidopsis thaliana to North America has been sort of well documented. There's a particular lineage that's established in North America something like 200 years ago, and using whole genome sequencing, this team could sort of reconstruct the mutation rate of in this lineage. And also the point of their work, I guess, was to demonstrate that these herbarium specimens provide a really nice sort of framework for natural experiments that have occurred, where a lineage is transplanted to a new area, and we have so many specimens from the relevant region that we can really have good resolution reconstructing the population genetic changes that have occurred. So they're good resources. My own work as a postdoc with Tom Gilbert in Copenhagen was to track the movement of a plant pathogen, the Phytophthora infestans and oomycete plant pathogen that caused the Irish potato famine when it was introduced to Europe in 1845. And to do that, we basically went right back to the source, to a potato plant that was collected in 1840, around 1845. I think it was 1842 for this one. And the lesions from the pathogen were on there, and we could directly sequence the genome of the pathogen and compare it to reference material from lineages of this pathogen that still circulate today. And surprisingly, the pathogen angle has been pretty important with herbarium genomics since then. There have been other works reconstructing historical genomes from, for example, 150-year-old citrus plant pathogens, and basically showing how these pathogens are moving around in the historical period, and how bacterial and fungal pathogens often have lineages that completely replace other lineages when they involve some kind of innovation. Continuing along this line, some of the early work on metagenomics in herbarium specimens was pioneered by Mikkel Schubert when he published the Palaeogenetics pipeline back in 2014. Basically included in this package was the capability to analyze the reads that are not mapping to the reference genome of interest, and then basically just doing a basic metagenomic analysis. And something he noticed early on was that in the few herbarium specimens that we had data from for these potato specimens, they were basically not all having exactly the same metagenomic community. There was some structure to them. You can see the two lineages. And I found that kind of fascinating, and I think he did too. We didn't know why that might be, but it opened up kind of a new world where we could say, well, is it based on particular genetic lineages of the host potato, or was it in fact a signature from the herbarium collections themselves? Was it like a local herbarium bacteria or something that was driving this? And he basically didn't pursue it, but since then, we followed up with some more work looking into those kind of questions. So the case study that I want to talk about today is on using historic herbarium specimens to study the invasion of plants, plant invasion. And the kind of questions we would like to ask about plant invasions are basically, how can we use these time-stamped, really nice samples from the herbarium collections to reconstruct these natural experiments where the plant has been introduced to a novel range and had to adapt to that range. And in some cases, an invasive plant is introduced to multiple ranges at the same time and has to adapt. So is it adapting in parallel? The same genes are being recruited to adapt to the different environments, or is it sort of like de novo mutations that in each case are being recruited for adaptation? There's some ideas out there that invasive species can either experience admixture in the native range or in the introduced range after they've been introduced to a new environment. There has been some controversy about that, so this is another thing that we wanted to pursue in our case study. And of course, something that I've always found fascinating, going way back into my PhD, was the idea that a species could become invasive if, based on this hypothesis that it escapes from enemies. They could be microbial enemies or macro enemies, like a herbivore, for example, for a pathogen from the native range. And when it moves to the introduced range, it's sort of like there's nothing there that's been co-evolved to fight against it. So it might have some ability to relax its defenses and move on to move those resources into reproduction, for example. So the case study I've been working toward introducing is on common ragweed, Ambrosia artemisifolia. It's a annual weed that grows and dies once per year, and native to North America. It produces highly allergenic pollen wherever it is. The pollen contains one of the most potent aeroallergens that there is, so it causes hay fever, and there's a global health burden for wherever the plant has large populations. It was introduced to Europe in the 1800s, and it was actually introduced globally. It's on many continents, excluding South America and Antarctica. And we know a lot about the history of common ragweed in North America from these sediment core records, the paleoecological records from all of the lakes that have been cored all over eastern North America. It has been well known for quite a long time that around the time of European colonization of a region of North America, like a local area, there's an increase in the abundance of ragweed, Ambrosia pollen. It's called the Ambrosia horizon, and it's a really good marker for when the landscape was deforested and switched to farmland for European colonists. So basically, we can see from these paleo records that that ambrosia was not very common in eastern North America, basically until European settlers arrived and deforested the landscape. We also know that after the plant was introduced to Europe, the populations experienced rapid adaptation. Basically, the plants that you find in Europe are larger than than the plants that you find in North America in the native range. They have larger investment in reproductive growth. And actually, the plants that have been introduced to North America since around the year 1800 have already developed latitudinal clines in flowering time and reproductive biomass. This is revealed through common garden experiments. So this is basically confirming that they have experienced this adaptive pressure since they arrived in Europe. Now the plant is distributed, of course, in its native range in North America, but also through much of Eastern Europe, where it's particularly an invasive and aggressive invasive plant, but also Western Europe. In France, it has been a major problem now for 150 years, and there's like more ephemeral populations established in Italy and Germany since then. So we wanted to pursue these questions about invasiveness using genomics from historic herbarium specimens. We first had to produce a reference genome in order to have something to map against. I will always regret that we didn't put a lot of thought into which plant we would actually generate the reference genome for, and we chose one that was incredibly heterozygous, possibly even like an outlier in heterozygosity. It was 2 to 3% heterozygous, and it's been a nightmare ever since for 6 to 8 years. We now have a much better chromosome resolved assembly, but for this particular work, we were still using the old one. So just to say, sometimes these decisions have consequences. All of the work that I'm presenting now is the work of my former PhD student, and she's now a postdoc with me at the museum, Vanessa Beaker. So all credit goes to her. That's Vanessa sampling one of her first ragweed plants like 6 years ago. So what we did was to generate a huge sample set to look at these questions, basically because we wanted to have nice spatial and temporal resolution of what was happening. And also because these plants are a weed, they're very abundant in the herbarium collections. There are usually stacks like this of the plant available in each herbarium. There's hundreds of specimens, and the curators are not particularly protective of the plants because it's just a weed with lots of material. So we did what we could. We generated 300 historic genomes from across the native and introduced range in Europe, as well as 350 modern genomes. And in particular, Vanessa did a good job to find places where we knew we had enough samples in a particular region in the historical period, and we tried to match with samples either collected fresh or that were already available in the modern period so that we could have a direct population sample to compare over time. So here we want to look at the change of allele frequencies in the same place over 150 to 200 years. And that's represented in these black dots here on the right side. So this is where all the samples came from. We processed all the samples in our clean lab at the Norwegian University of Science and Technology. We did everything high throughput using single tube library preps, where we could basically build libraries in plate format. And we did our best to keep the amount of sequencing as low as possible in order to still infer what we wanted to, but spend as little money as we could, basically. So then we used genotype likelihood approaches, which are rather common with ancient DNA. And what we find is that if we just look in the historical period in North America, there's some pretty clear genetic clusters that are formed. Here, there's highlighted 3. There's an admixture zone, which is basically between the other 3 sort of clusters, and we associate that with this disturbance, human European colonist disturbance of the landscape, basically mixing and moving the germplasm, the seeds back and forth between the population, between the regions in North America. And we see that in the modern period, basically we have exactly the same genetic structure. So although there's been an increase in the population sizes, there has not been a big change in structure itself. On the other hand, if we add in the European samples from the historic period, we find that they are occupying just a subset of the diversity of the native range, and so it basically shows that the material introduced to Europe in the historic period was basically only coming from certain parts of North America. This was an open question at the time. And in the modern period, we see something happening where there's particular population locations that have diverged very much from the other populations that we surveyed. And you can see them here forming outliers on the PCA, sort of like very far away. So here we're interpreting either rapid adaptation or introgression with outgroup species, which we have a bit of that discussed in the paper, but I won't mention it anymore now. But basically, we took interest that there's some places where we're seeing very, very strong changes in the allele frequency over a short period of time. Vanessa basically did some selection scanning using all these genomes. So in this approach, we looked at, we scanned across the genome for very large changes in allele frequency between the historical and modern European population using FST, and we used some other measures too. And we looked at what kind of genes were in the regions that had been where the allele frequencies were highly altered. And basically, we can summarize that it was some things that we would have expected from the previous work, showing that rapid adaptation had occurred in Europe, like the flowering time genes, for example, were particularly represented, as well as adaptation to different environments that were more or less arid and saline. But we also noticed that one of the top Go terms that was represented here is about defense and innate immunity in the host. So then we became interested in that, and we decided to follow up on some previous work Vanessa had done looking at the metagenomic composition of historical herbarium specimens, in which basically we are taking all the reads that don't map to the reference genome and then just doing a basic survey of what that kind of diversity, what that microbial diversity is. And I guess what Vanessa's innovation was in her initial work was to take a comparison of one type of plant, one species of plant, and another species of plant, Arabidopsis thaliana and Ambrosia artemisifolia, and to look for the microbial taxa that were represented in both in the historical samples, and then sort of remove those from the analysis. And what we inferred there was that this was post-mortem colonization of the plant materials basically after collection, which then allows us, if we can remove those, we can then look at the true nature of the historical metagenome. So Vanessa tried that approach, and particularly she wanted to look at the pathogens that she found on the leaves to test this idea of enemy release. We didn't see much there. It was pretty much the same number of pathogens present in Europe and North America. But what she did see, there was a big difference in particularly this bacterial genus called Xanthomonas, which are obligate plant pathogens, and they're usually very host-specific. She saw basically that there were a lot more abundant types of Xanthomonas in the native range than in the introduced range, which we took to be some sign that something was going on with Xanthomonas. Maybe escaping from Xanthomonas was part of the story here. So just to sum up what you can read more about it in Vanessa's paper, but basically we found rapid adaptation in particular types of genes. Some of those genes were related to defense, and we saw some sign that there could have been an escape from enemies in the native range. And there's some other caveats and other things that are discussed in the paper, which you can hear about. The next steps are to fix the problems with basically we see some temporal biases in the metagenomic signatures. It seems like there's always a big shift over time in what microbial communities are present, either in North America or Europe, and we're not quite sure we believe them. I think it's based on the length of the DNA sequence reads. We're going to follow up with complete metagenomic assemblies so that we can look functionally at what type of genes are represented in the historical and modern Xanthomonas genomes present on these plants, and we are going to look for associations between the host genotypes in these populations and the microbial community that they support. So that's the next steps for Vanessa. With that, I would like to thank, first of all of the herbarium collections that contributed to the project. You can see there's quite a few museums here. I think there's something like 20 collections, and the curators are really nice to allow us to destructively sample. Very many co-authors and contributors to the work, and the research group, holo-genomics, back in Norway, has always contributed as colleagues and friends. So thank you, thank them.
Thanks, Mike. That's really a fantastic presentation. We have to invite you back for seminars to talk about your other research projects at some other time.
Sure, I'd love that.
I think it's also branching out, just showing that the plant analysis is, in herbarium and also ancient plant material is definitely something that is very valuable.
Yeah, and there's a lot of ancient plant projects coming up. It's just I think not many of them have come to fruition yet. People are looking at the domestication of plants going way back. So.
Right. So maybe one question before we move on, for the sake of time. Does anyone? One question from the audience? All right. Otherwise, we can grab Mike after the meeting and ask any questions you like.
Thank you.
All right. Thank you, Mike. All right. Moving on. So next speaker is Marianne de Hask. She's going to talk about optical duplicates, the effect of loading concentration on library complexity. So a technical but very important issue. So please go ahead, Marianne.
Okay. Hello, everyone. My name is Marianne. I'm currently actually employed as a lab manager at the Center for Palaeogenetics. But I'm also still a PhD student. And during my PhD, I'm mainly studying the woolly mammoth. We've already talked a bit about mammoths today, but I'm mainly focusing on one of the last woolly mammoth populations. There was a population that lived on Wrangel Island, which is a small island in northeastern Siberia. And their mammoths became isolated about 10,000 years ago when the sea level started to rise, climate got a lot warmer. But then on that island, they actually managed to survive for about 6,000 years before they eventually went extinct. And one of the main questions I'm trying to solve, or I'm asking myself during my PhD, is what kind of genetic processes were there at play before the mammoths eventually went extinct? And so here you see me posing with probably the most important sample we have in the lab. It's so important that we even give it a little nickname. It's called Lonely Boy. It's a reference to a famous TV show, but it kind of also makes sense. This sample has been dated to 4,000 years before presence, and with that it is also the youngest sample as far as we know that has actually been dated. So for my question, understanding why mammoths went extinct, what happened on a genetic level, you can imagine that this is quite an important sample. And so we sequenced this sample a lot, like really a lot. But we had low complexity in this sample. And what does that mean? We would take a step back and we just start by talking about sequencing duplicates. So when we extract DNA from our mammoth and then convert it into libraries, we'll talk a lot about those. Those are basically extracts ready for sequencing, very easily put. And then we puzzle them against the reference genome. You see that one here. And what we then sometimes encounter is that we have fragments that have exactly the same size. They map to exactly the same location. They might even have the same mutations. So those are sequencing duplicates. And they're a problem, because these duplicates, they all have the same source. They might bias downstream analysis. We might think that we have way better coverage than we actually have. So when we sequence a lot of duplicates, we actually don't get a lot of new information in our data set, and therefore they inflate sequencing costs. We don't want that. Sequencing is still quite expensive. So what is the source of these duplicates? If they are such big a problem, why don't we just, find ways to not have duplicates in our data set? Well, the source of these duplicates, or the main source, I think what most people think about, are PCR duplicates or library duplicates. So we start from, oh, this is going to be tricky. Okay, so basically on the left, we start from a single DNA molecule, to which we basically attach two blue, let's call them adapters, which simply put will allow this DNA molecule to bind to the sequencing flow cell, so which allow the fragments to be sequenced. And while doing that, we also amplify the DNA to increase the concentration. as far as I know, or at least in our lab, this is something we do for every ancient extract. So what does it mean when I say that Lonely Boy, my favorite sample, has low library complexity? It basically means that it has a very small fraction of unique reads. When we sequencing Lonely Boy, we are getting a lot of duplicates. And you can see the results of that here. I sequence Lonely Boy a lot, so we have a lot of data on it. What you can see here on the figure is on the x-axis sequencing effort, so basically how many million reads we sequenced per library, and on the y-axis the library complexity. And as expected, the more you sequence the library, the more duplicates you will encounter in that library, so the lower library complexity gets. Seems to be a pretty straightforward pattern. But there's something weird about this data. If you group them by sequencing run, another pattern becomes evident. So this sample was sequenced in 3 different sequencing runs, and it seems that something else is driving the duplicates here. We see that for the red sequencing run, or the dots on top, is that they seem to have consistently lower or duplicates or higher complexity compared to the yellow and the blue run. And this has been puzzling me for a very long time. What's driving these duplicates? Is this me screwing up something in the lab? Is there maybe a difference between sequencing machines? We changed sequencing from HiSeq-X to NovaSeq. I looked whether maybe the fragment size of the sequencing could be involved, and I just could not wrap my mind around this. Because I didn't only see this pattern in Lonely Boy, I saw this in a lot of samples. And then suddenly, I think it was someone on Twitter mentioned optical duplicates. I have sequenced a lot, and I had never heard of optical duplicates before. But so basically, what are they? So for this, we go back, or we take another step back, and we look at what Illumina sequencing, what actually happens when you do that. For this, I actually brought a little prop. They generously gave it to me outside. So how does it work? You can maybe see it. This is a flow cell, and this flow cell has 4 lanes. And so what happens is when you load this flow cell is that you load the DNA on one lane, and then the DNA will attach itself to the lane, to the flow cell. And then there are different scenarios. You can have under-clustering, as shown on the left. You can have optimal clustering and over-clustering. And so one of the problems with, for example, under-clustering is that this will artificially generate optical duplicates in your data. So what is a cluster? A cluster is basically a DNA fragment that binds to the flow cell, then it becomes a cluster, so it again duplicates itself. So each cluster represents one sequencing read. But what might happen in those under-cluster flow cells is that one cluster by the software, by the machine itself, will get recognized as 2 clusters. So once you get your sequencing data, the software will tell you, like, hey, these are 2 independent reads, whereas actually they belong to the same cluster. So again, introducing duplicates in your final data set, which we don't like. So you might think, well, easily solved. Why don't you just always aim to over-cluster your run? But that's over-implies. It's not very good. I think the main problem associated with that is that over-clustering also tends to reduce sequencing quality. So yeah, sure, you might sequence more fragments, but the quality might be lower. So it's not sure whether you actually get more results from that. So for this, we actually had a postdoc, Nick Dussex, who was also very annoyed by this problem. He reached out to SciLife, and they were then so generous to offer us actually a sequencing experiment. So they said, if you give us the libraries, we will load them at different concentrations on the flow cell I just showed. So every lane would have a different loading concentration. So basically we test what's the effect of under-clustering, what's the effect of optimally clustering or over-clustering, or maybe we're not over-clustering at all. So there were 4 different loading concentrations, 500 picomolar, 400, 300, and 200. On the right, you can see how many clusters or how many sequencing reads we had at the end of every sequencing run. And I think, if I'm not mistaken, or most of our samples are currently run at the 200 picomolar. So that's good to keep in the back of your head. And what kind of samples did we provide? Well, we tried to mix it a little bit. So we gave them 3 relatively young, late Pleistocene mammoths. So these are less than 50,000 years old. 2 are pretty good samples. 1 is a low complexity sample. So same issue as Lonely Boy. We know there are a lot of duplicates there. And then we also borrow 2 mammoths from Camilo, middle Pleistocene mammoths. So those are very old. We basically expect them to be extremely bad. So SciLife sequenced the data for us. We then processed the sequencing data with the GeneRoad pipeline, which is a pipeline that was published earlier this year, which allows simple processing and analyzing of ancient data. And then we used a tool called SumTools Markup to label the different duplicates in our data. So label them either as being an optical duplicate, or a sequencing duplicate, or a library duplicate. And the way the software generally does it is by looking at the distance of the duplicates on the flow cell. So if they're very distant apart, the software will label them library duplicates. If they're very close together, the software will label them optical duplicates. So first results, this is from one of the well-preserved mammoth samples. On the x-axis, you can see loading concentration, which goes from high to low. It's an important one to know. And on the y-axis, you see the ratio. So the ratio basically expresses for every library duplicate, how many optical duplicates do we see in our data. And we see a pretty clear pattern here, which seems to be that the higher the loading concentration, the lower the amount of optical duplicates per library duplicates. And we see this sample in all our late Pleistocene mammoths. So you also have the low complexity library where you see the same pattern. And then the other well-preserved sample, there is one weird outlier at 500 picomolar. I have no idea what that is. Maybe it's me that made a mistake somewhere, or maybe it's the software mislabeling. So maybe just looking at the ratio optical or library duplicates is not ideal. So then we looked at a second parameter, which is coverage. So coverage meaning for every position in a reference genome, how many times independently is that covered by another read. And coverage, I think it's a very good proxy for sequencing efficiency, because it depends both on the quality of your sequencing data, it depends on the amount of duplicates, it depends on fragment length. So it really looks at all those things that play together. And so on the x-axis, again, loading concentration from high to low, left to right, and then coverage on the left. And I think this is, again, a pretty clear pattern. It's actually a significant pattern where we see that the higher the loading concentration, the more coverage we get. Now, again, this is a very well-preserved mammoth. What does it look like in our other mammoths? Well, basically the same. Even for our other well-preserved sample that had that super weird outlier at 500 picomolar, even though there's a lot of variance, for 500 and 400 picomolars, it still has significantly more coverage than for the other loading concentrations. And even if we look at Camilo's very ancient mammoths, the one on the right, middle and bottom, even they show the same pattern. So overall, it seems that there is a significant difference between loading concentrations, so they do indeed affect the output of the data. That's maybe not a surprise. But most importantly, it seems that higher loading concentrations are actually more beneficial for our ancient data set. So yeah, with that, I would like to thank you all. Ooh, spoiler.
Thanks, Marianne. I think that's excellent work, and that's something that's really valuable for us actually, spending time in the lab and preparing this material for sequencing and generate the best possible data. So, any questions for Marianne at this point?
As Magnus said, I was just wondering. So it seemed that line hasn't flattened out at the top. So
yeah.
do you think by increasing it even more, it it might get even better?
Maybe, yeah. I mean, I think like the main consideration we had with these higher loading concentrations was that we would see a drop in quality. But even if we filter for quality, it keeps going up. So yeah, maybe there could be a second experiment. But I think even if we just use this could already generate a lot more data, usable data.
Thank you. What's the default used currently?
I'm not sure, but I think it's 200, so the lowest one in this experiment. But I'm actually not sure, so maybe someone at SciLife who actually did the work could comment on that. Okay, 300. That's what they said in the back of the audience.
Any last question? All right. Thank you, Mariana.
Yeah.
All right. So have reached almost the end. We have saved the best for last. Tom van der Valk is going to talk about identifying admixture and ghost lineages in ancient genomes, particularly. So we are very happy to have you here. So please, Tom.
Yes, seems to be working. So before I started, I just want to thank you, Magnus, for putting together such a nice symposium today. I was impressed by the diversity of the speakers. I really enjoyed it. So very nicely done. And thank you for giving me the opportunity to talk. I'm going to talk today about humans again, so human genomics. And I will talk a bit about Neanderthals and Denisovan, which I think is very fitting given that the Nobel Prize, this symposium was organized before the Nobel Prize was known, but I think now that we know it was given to Svante for his work on Neanderthals and Denisovan, it would be unfortunate if we don't talk a little bit about it. I'm also going to give a caveat. There are several people here in the audience who know way more about human genomics than I do. I mostly work on non-human animals. But I'm very interested in so many of the method developments, the bioinformatic and computational tools we're using are actually driven by the human genomics field, and I'm very interested in applying those methods to other species. So I will mostly talk a little bit about the methods part, and in the context of what we can actually learn from modern genomes, which are very powerful, and how can we use the statistical power of modern genome to infer past events on human history, but also other species. Here I'm showing a very simplified model of our evolutionary history. And I think these two events here are interesting. So the Neanderthals and the Denisovan, where we see some mixture with Homo sapiens. How do we know that this has actually occurred? Well, there were some hints in more recent papers, but I think these two papers were highly influential in the fine-scale mapping of when this gene flow occurred, how much gene flow occurred, in which human populations did it mostly occur, in which human populations it didn't occur. So this was the first high coverage, complete Neanderthal genome. And then in the same year, if I say it correctly, there was the high coverage Denisovan. So that was a human lineage that we actually didn't know existed. So we more or less know about this human lineages from the genetic data, not so much from the fossil record. And how then do we actually know that humans, so modern Homo sapiens, has admixed with these Neanderthals and Denisovans? Well, there are many fancy statistical methods, but in the end, they all boil down more or less to the same kind of analysis, which we often refer to as D-statistics or Abba-Baba test. And the way it works is as follows. So imagine we have several genomes, and for this test, we need an outgroup genome. And in human genomics, we often use, or in all cases, we use the chimpanzee genome. And this will allow us to look at the ancestral state of an allele, so we know how the sequence of that allele was in our very deep past. Then we have, for instance, in this case, an archaic human, so this can be a Denisovan or a Neanderthal genome, and we have different human populations, which we refer to as the P1, P2, and P3 groups. Now you can imagine if there is a mutation that happens in the history of
shared history of archaic human and modern human population, several things can happen. First of all, that mutation can end up in all the populations. So we can find it in, imagine we have an African population, Eurasian population, and we have a Neanderthal. We can find that we are identical at this specific part of the genome. So that doesn't really give us a lot of information. We are all identical. Something else can happen. It's called incomplete lineage sorting. So a certain mutation can end up in an archaic genome and in one of the two human populations. The same pattern can happen the other way around. So the mutation ends up in the archaic human and in the other, in this case, the P1 population. So we expect on a genome-wide level, given that the genome has billions of sites, we have a lot of statistical power. So if we do this for the whole genome, we expect this pattern to be completely equal if there is no gene flow between any of the populations. However, in the scenario that we actually have gene flow from archaic human into a specific human population, but not in the other, if we then just very simply count how often do for instance, Neanderthal and the P2 population share a genetic mutation that the other population doesn't share. So we count how often do we find this composition, how often do we find this composition. Then you will find under a scenario of gene flow that these two share more often a genetic variant than expected from a random pattern without gene flow. This is just very simply counting the number of allele sharing, and then you can use a lot of fancy statistical models that we have seen earlier today about all the human migrations, human gene flow, and you try to model different patterns that match the counts you actually find in the genomic data. So that's how we infer and know about these admixture events, and we can even estimate how much gene flow was exchanged between archaic humans and modern human populations. This method also has some disadvantages. So as I showed you earlier, we have an outgroup genome, and then we have the archaic human genome, where we do the counts of allele sharing between different populations. Now, what if you have a human population that, or an archaic human species that has existed in the past that we don't know about? For instance, the Denisovan is I think, a cool example. The Denisovan was kind of a surprise that we found it, and a few decades ago we had absolutely no idea that this human existed. And there might be more of those cases out there where we just haven't found a fossil that we can obtain DNA from, so we don't know that this unique lineage existed. Well, we can actually use the power of modern genomes to look for patterns of these extinct lineages, which we now in the field often refer to as ghost lineages. So we do not find them anywhere alive today. We don't find them in the fossil record, but they might still be living as a part of our genome, in leaving a genetic trace in our genomes. So how do we discover these ghost lineages? This is one slide about the method. I hope this won't be too boring, but I try to explain it a little bit simpler. So this is actually relatively new research. So these methods have been mostly developed in the past few years, and one of the a very popular one is by Lauwitz-Skoff et al. And this works as follows. So again, think at the same tree as I showed earlier. We have, in this case, an outgroup population, so that could be, again, be a human population. We have another human population that has admixture with an archaic species. But then keep in mind, we don't have the genome of this archaic species. So what happens during an admixture event? Well, you can imagine that these mutations that are unique to this archaic human now also end up in your population that received gene flow from this human, whereas they are not found in an outgroup that didn't receive this gene flow. So what you can then do, if we look at specifically as this in-group population, we can look at all the genetic variations, across the genome. So we do that in Windows. We look at all the genetic variants, and then we can remove all the variants that we also find in an outgroup that we do not expect that has received this gene flow. And then we can simply count in the different windows how many unique variants do we end up with after we removed all shared variation between two populations. You can then train a hidden Markov model on this, and you run the hidden Markov model back basically on your counts, but here you also take into account recombination rate and mutation rates of different parts of the genome. So different parts of the genome mutate at different speeds, so you have to account for that when you do these variant counts. And then you get probabilities of that a window is likely just from, in this case, humans, or whether it might have been inherited to an archaic gene flow event. So if you train the model and run the model, so here you can see that windows with little variation are very likely human, and windows with a lot of unique variation are classified as archaic. So you can run that on your counts, and then what you get is for each window you get a classification, either being your in-group, so human, or being from an archaic unknown species, and you get likelihood values for each of the windows, so you can do downstream filtering of how confident you want to be about these inferences. So what can we do with this method? I'm going to give a few examples that. So I haven't been involved in any of these studies, but I think they're super fun. I really love reading the papers, so I'm just going to show you some examples of how you can use this method. So the first paper by a large Icelandic consortium. In Iceland, they sequenced many Icelandic genomes, and some modern genomes, that is, and you can do really fun stuff with it. So this is Hans Jonathan. He was born enslaved in the Caribbean at the around 1780, and he had a Danish father and a mother from African descent who was brought to the Caribbean as a slave. And his owners were Danish, and during his early puberty, his owners moved back to Denmark and took Hans Jonathan with them, where when he was 17, he escaped, joined the Danish Navy, fought in the Battle of Copenhagen in 1801, and that gave him, the crown prince back then in Denmark gave him his freedom in return for fighting in this battle. Then a famous lawsuit happened where his previous owners claimed that he still belonged to them, and they won the lawsuit. So he was back enslaved again and escaped another time, and this time to Iceland. And so he's the first known immigrant in Iceland from African ancestry. In Iceland, he got 3 offspring, of which 2 survived, and those 2 surviving offspring got more offspring again. So here you can see a family picture with his sons and grandchildren. So it was unknown where Hans Jonathan, his mother, came from. So what they did in Iceland, they sequenced. They sequenced his known descendants, and then they used this method that I showed you earlier of identifying unique variation in windows to find parts of the genome where, so these are all Icelandic people, and they have a very large reference set of other Icelandic genomes that are not descendant of Hans Jonathan. And then they could identify these windows with a lot of unique variation. They run the model, and then in blue here you can find, so each yellow bar here is an individual, and you can find in blue the parts that are sticking out. And because they sequenced all his descendant, they could actually reconstruct 36% of the genome of Hans Jonathan without ever having sequenced himself. So what can you, so here again, they could then reconstruct 36% of Hans Jonathan's genome. In blue, you can see the parts that they could reconstruct from all his offspring. And then what you can do is you can take only those blue parts and plot them on a PCA plot. So we have seen several PCA plots today in the context of human ancestry. So here's the PCA plot they made. So this is a data set of African genomes from different, across Africa. You can see here the correspondence of the colors. And when you then take these blue parts of the reconstructed genome and you plot it on the PCA plot, you see that the mother of Hans Jonathan came from the area that now spans Benin, Cameroon, and Nigeria. So now we have reconstructed the ancestry of the mother of Hans Jonathan without ever having sequenced any ancient DNA. So I think this is super fascinating that we can do that from just modern genomes. Another example from the Icelandic consortium again. This time they sequenced over 27,000 complete genomes from Iceland, and they compared that to a large data set of African genomes to basically look again at these windows that have a lot of unique variants that are not shared between Icelandic people and African genomes, and then again looked for all outlier windows that might be a signature of archaic introgression, so part of the genome that have an archaic signal. If you summarize that over all the Icelandic genomes they used in this study, it looks a bit like a complex plot, but here you have all the chromosomes again, and then the different colors represent. So after they identified all these windows that seem to be from an archaic introgression event, they then matched how closely are these windows to known ancient genomes that we've sequenced. So the Altai is a Neanderthal, the Vindhyi is a Neanderthal, and then we have the Denisovan genome, so we can look at each of these windows after we have found them, so without using any of this information, what are they most closely related to? And you get a map of Neanderthal and Denisovan introgression in the Icelandic population. So the first thing they find is that Icelandic people have Neanderthal ancestry, which was already known, but they also show that it's more closely related to this Neanderthal genome than it is to this Neanderthal genome. So it's mostly the blue color, and you find little relatedness to this. And then also, maybe somewhat surprising, they actually find Denisovan ancestry in Iceland, and Denisovan ancestry is thought to be mostly in Southeast Asia and Oceania. And then, so they modeled this kind of the statistics, so the relatedness between the Icelandic people and these archaic genomes. And what they come up with as the most plausible hypothesis is that actually that Icelandic people got some admixture gene flow from a Neanderthal species that is, or actually I shouldn't say Icelandic people. This goes for all non-African genomes. But this study was specifically done on Icelandic genomes. So they received gene flow from a Neanderthal closely related, or relatively closely related to this Neanderthal genome, so not from the Altai Neanderthal, but from this Neanderthal genome. But they hypothesized that actually Denisovans contributed gene flow to this specific Neanderthal, which then contributed some ancestry to non-Africans, and that's why we find Denisovan DNA in people in Iceland, even though there very likely was no direct gene flow from Denisovan into this population. And again, this we all learned from a very large data set of modern genomes, and we can make all these archaic inferences. A final paper about humans is this paper, which uses a similar method as the one that I described earlier. It's called S prime, or an updated version of it is S star, where they looked at specifically Denisovan admixture in Oceania and Southeast Asia, where we know most of the Denisovan ancestry remains today. They looked at over 5,000 genomes, so genomes from both Oceania and African genomes, and again looked at this private, unique variation in the genomes, and then identify all the windows that that seem to be coming from an archaic human. So what they then did with the window is here, they took a British genome, and they plotted on the x-axis, they have the distance to the Altai Neanderthal, and then the y-axis, they have the distance, genetic distance to the Denisovan. And then they plotted all these windows here. And what you can see is that, so this is a density plot, and the peak of all the windows in the British individual is, has very little genetic relationship to the Denisovan, and it shows around an 80% relationship to the Altai Neanderthal. So this shows you that British people, or this British genome, has Neanderthal ancestry, and it was somewhat closely related to the Altai Neanderthal, but not a perfect match. If you then look at a genome from Papua New Guinea, which we know is the population today that has the most remaining Denisovan ancestry of all human populations, or among the most Denisovan ancestry. We again see here the Neanderthal. So this is the same, they plotted all the windows. We can see again the Neanderthal signal. So that's the same signal as that the British genome carries. But then we can also see this very strong signal of Denisovan ancestry in the modern Papuan genome. And here you can also see that these windows are actually, they are Denisovan, but they are not closely related to this Altai Denisovan. So it was a Denisovan population. It was a different Denisovan population than this specific Altai Denisovan, even though we know it was Denisovan. But then the most interesting finding, I think, and this is, this again shows you the power of modern genomes. So this was completely discovered using the modern genome data without having actually all the archaic genomes available, is from East Asia, specifically here, southern Han Chinese. Again, the same Neanderthal ancestry, so we know that all humans outside Africa carry the same Neanderthal ancestry signal. They also have the Denisovan signal that the Papuans have, so you can see the around 60% sequence match to the Altai Denisovan. But then they also have a second pulse of admixture, which is very closely related to the Altai Denisovan sequence we have. So the authors of this study concluded there was not a single Denisovan admixture event, but there have actually been two major pulses, one very strong pulse in Oceania, and then another pulse in East Asia of Denisovans that were very closely related to the Altai Denisovan of which we have the genome sequence. So I think this is super cool that you can find like a new, you can learn like a new migration pulse from Denisovans just from looking at the modern genomes. Finally, are we humans so special that we hybridize and mix all the time? No, we're absolutely not. Here's two papers about chimpanzees that are using similar methods. This one, again, based on modern genomes, showed that chimpanzees have separated from bonobos around 1 million years ago, but then identified later admixture events. This was the classical D-statistics that I showed completely at the beginning of my slide, but then also a paper by Martin et al. He then used this Lowitz-Skoff method to look at signals of ghost introgression in chimpanzees and bonobos, and they didn't find any of such pattern in the chimpanzee, but actually in the bonobo, they show that the bonobo also has this signal of ghost introgression. So there was some kind of common ancestor between chimpanzees and bonobos that diverged, became its own lineage, and then later contributed some of its genes to the bonobo population. Here I'm just showing this is how the different chimpanzee subspecies are related to each other, and here's the bonobo, and this is a relative scale of genetic distance between the different species. And then when you take the windows, so they only discovered a pattern of ghost introgression in the bonobos, and if you take those specific windows that show this signal of ghost introgression and you draw the same tree, you can all of a sudden see that the genetic distance, as you would expect, is much, much larger. So this is a species that was quite highly divergent from the chimpanzees and the bonobos that has contributed some of its genes to modern-day bonobo individuals. And so this is the model that they come up with, and as you can see, as with humans as well, there has been gene flow throughout the chimpanzee evolutionary history. But then there's also this signal into the bonobos from a quite deeply divergent pan species, so between 2 and 3 million years divergence from modern-day chimpanzees and bonobos that has left a signal in the bonobo population. Finally, I did my PhD mostly on gorilla genomics. Unfortunately, I cannot show results about our ongoing work on this, but we are looking at ghost introgression and gorillas and writing up the paper right now. And as you can expect from the previous examples I gave on chimpanzees and humans, you can guess that it's probably similar things going on in the gorilla populations. Thank you.
Right, was a really spectacular talk, and thanks for those insights into how to combine modern ancient DNA and sort of computational analysis, find these ghost populations. So we have one question in the back.
Firstly, thank you so much. I just love any story that involves integration, just simply because I feel like we often operate within a paradigm where it's the exception rather than the rule, and what all the papers that you've demonstrated, and it seems like everybody who's published anything in this room, especially presented today, is that far from being the exception to the rule, it is the rule. And that seeing all kinds of admixture everywhere and our ability to actually detect it is just absolutely outstanding. And I hadn't known about some of these other examples, so it's just super exciting. The only thing that I would say is I think we just need to be sometimes a bit careful about our language, and I'm aware that there are populations of humans who hear the word ghost and have a particularly negative response to that, and I'm thinking that there are maybe some of us in the room, if we'd referred to those populations as either zombie populations or any other sort of monster thing that might not, because ghost I think for us doesn't mean, for a lot of people anyway, it maybe doesn't mean, it's kind of a neutral word. It doesn't mean, you can think of Casper or something, it's kind of fine, but for other populations it's not so much, and I think that we're moving towards saying unsampled populations or something like that, but I appreciate there's conversations around that, but I just wanted to raise that as for like food for thought, and maybe to get your response on that as well.
Yes, no, I think I actually agree with you. I don't work on human genomics myself, and part of the reason is that there is a lot of ethical complication that I don't, I'm not the right person to talk about or think about, yes. But I think I agree with you that neutral terms are better. This is the ghost admixture is not the term that I came up with. It's a term used in the literature, and maybe like we say it to all the people here working on human genomics that maybe think about this term. Maybe we can move towards another term. Yes.
Like old world, new world, right? I mean, there's similar kinds of things about the terminology we use and how that reflects a kind of particular
absolutely, and it has changed in the literature over time. So yeah, I think it's always good that the field is aware about it. Yeah.
Thank you.
Thank you, Tom. It's super interesting. It's super fascinating to detect this ghost introgression or admixture events. I just have a problem with validation. So is it possible, just to take a modern human population with a known family history and pretend that we don't know who the ghost is, that has some ghost lineage, and we pretend that we don't know that there is a ghost lineage. Sorry for using this language. But and to sort of reconstruct this unknown lineage. Because I assume all these hidden Markov models, they're trained on simulated data. But do we have sort of concrete examples? For example, about the tracing the ancestry of the mother of the HJ. Sorry, I forgot the name.
Hans Jonathan.
Yes, Hans. This guy. Was it like confirmed from family records that his mother was from this part of Africa? Like, how much can we validate all this inference of introgression?
Yeah, I think part of it comes back to, as you say, the simulations. So we know the methods work from simulations where we know the ground truth. And then the biological data always becomes way more complicated than the simulated data. I think a few of the examples that I showed you here is they identified windows that have an archaic signal, and then they compare it to the Neanderthal or the Denisovan known genome sequences, right? And they show a match there. So in those cases, I think you have a validation there. It would be, if the method would just pick up random windows, you would not find this genetic relationship. Now, there are several papers on human genomics where they claim introgression from unsampled populations. We saw an early example in Africa. There are some other examples as well. I would say the field as a whole is relatively skeptical about those as well. Like how in the end, how do you really validate that? In the end, I think you want to have the actual genome sequence of the introgressed species, which might not always be possible. But I think a few of the examples that I gave here show that the methods overall seem to work. Again, by comparing it to known archaic genomic data.
All right, thanks. Was there a last question over there? Yeah. All right, one final question there. Perfect.
Great talk, thank you. And also, so I'm very interested as you reviewed that from human modern population, you can infer the extinct lineage. And if applies to, for example, chimpanzee or other primates, like non-human species, how large is the sequence, the population size, or the amount of the alias that's required to actually detect an extinct lineage?
Yeah, it depends on a lot of factors. In general, more genomes is always better. That's why a lot of these studies are done on humans, where we have a lot of reference populations to work with. It depends on how divergent your unsampled lineage is from the group you're specifically studying. So the wider the divergence, the more unique genetic variation will be brought into your population. So you can detect very deep diverged lineages with much fewer genomes than, for instance, this Hans Jonathan example, where you need like thousands of Icelandic genomes and then the offspring of Hans Jonathan as your in-group. So I would say mostly more genomes is always better. In the case of the chimpanzee and the gorilla, we're working with tens of genomes instead of thousands, and we can still find those signals. But if you would have let's say a gorilla unsampled lineage that is very closely related to modern gorillas, you would not pick it up with that few genomes.
Does it mean that it's better to sample the genomes like in different geographic locations so that they are more divergent?
Again, it depends a bit on your question. Going back to the Icelandic sample, actually you want specifically to sequence Icelandic people that did not came from, that were not offspring of Hans Jonathan, and compare those with the offspring of Hans Jonathan, because then you can filter out all variants that is shared across Iceland. And in other types of questions, you actually want to sample all the genetic diversity in that species for filtering the variants. So yeah, it depends a bit on specifically what you're interested in.
That's very clear.
Yes. Okay, thank you again, Tom. So we come at the end of the meeting. I would just like to again thank all the speakers at the meeting for your fantastic presentation. It's been an incredibly rewarding day for me. I hope you have also feeling enthusiastic about the way this research field is going. It's a dramatically and quickly expanding field that will be no less now with this year's Nobel Prize to Svante Pääbo in medicine and physiology. It is a very expansive field, so I think for young researchers, students perhaps in particular, there's going to be a lot of recruitments coming over the coming time. So if you like to consider working in this field, I think there's going to be plenty of opportunities. Otherwise, I thank everybody who coming here to attending, both here on site and also online, for your participation, for your questions, I hope you had very fruitful discussions in the breaks as well. So some of these presentations will be able online. Some of these speakers included unpublished material have selected to not have their presentation available on YouTube afterwards, but most of these presentations will be available online afterwards if you want to come back to review them. Otherwise, yes, thank you for today, and hope you have a nice journey home. Thank you.